So good morning and welcome to the next in the series of highlighting organic chemistry across Europe. And I'm delighted that today we're highlighting organic chemistry in Austria. So without any further ado, I'll pass over to Michael Schnurch, who's going to give you an overview of organic chemistry in Austria. Michael.
Good morning, everybody. I wanted to give a quite brief introduction, but then actually I found out that there's quite a bit to say about organic chemistry in Austria. So let me start. Where are we? Of course, in the heart of Europe. Our Austrian Chemical Society, or as we call it, Österreichische Gesellschaft für Chemie, is headed by Professor Peter Gärtner and actually contains 4 working groups, which are either carrying organic chemistry in their name, as the working group of organic chemistry headed by Mario Vasa, one of our speakers today, or are quite closely related as the working group of medicinal chemistry, the working group of macromolecular chemistry, and also the working group of carbohydrate chemistry. I've marked here in Austria the universities actually which also are involved in the field of organic chemistry, and I will briefly introduce them from west going to the east. And therefore, we start with the University of Innsbruck. There we have a dedicated institute of organic chemistry with the colleagues Ronald Mikura, Thomas Magauer, Christoph Kreutz, Katrin Reuker, Thomas Müller, and Martin Tollinger. But we also, or they also, infiltrate the Institute of General Inorganic and Theoretical Chemistry. And our speaker today from Innsbruck will be Fabian Dillmann, from the working group of molecular inorganic chemistry. Let's say whether the "in" we can put rather in brackets, and I'm very curious what he's going to tell us today. And moving further to the east, we are at the Johannes Kepler University in Linz, and you see there the organic chemistry activities are split over 3 institutes. The Institute of Organic Chemistry with Wolfgang Schöfberger and our today's speaker from Linz, Mario Vasa, who is working in the field of asymmetric catalysis and synthesis. And we have the Institute of Catalysis with Marco Hapke and Christoph Topf, and the Institute of Chemical Technology of Organic Materials with Christian Paulik and Clemens Schwarzinger. Then moving to Graz, I don't know exactly which of the 2 universities there is more to the west and which one is more to the east, but let's start with the University of Graz, Karl Franzens University of Graz to be exact, where at the Institute of Chemistry there is one dedicated research division, organic and bioorganic chemistry, consisting of Katalin Barta, Thomas Glasnow, our today's speaker Melanie Hall, Oliver Kappe, Wolfgang Krutil, and Klaus Zanger. Then the second university in Graz is the Technical University of Graz with the Institute of Organic Chemistry and Wolf Breinbauer, today's speaker from Technical University of Graz, and Rita Fürst, and the Institute of Chemistry and Technology of Bio-Based Materials, where Tanja Vodnik is holding the flag of organic chemistry high. And last but not least, the Institute of Chemistry and Technology of Materials with Christian Slugovc and Gregor Trimmer. Then the University of Agricultural Science, Organic Chemistry at OKU, as we call the university short, also carries an Institute of Organic Chemistry with Alla Zamiatina and Fabian Prengle, who will represent BOKU today. Furthermore, the Institute of Chemistry of Renewable Resources, where Thomas Rosenau is quite involved in organic chemistry. Then our newest addition actually to organic chemistry, maybe most of you don't know about it yet, the Institute of Science and Technology Austria finally has hired chemists, and 2 of them are organic chemists, Rafael Klein and Bartholomeus Bieber. Unfortunately, neither of them could make it today. But I hope at one of the next editions we can then also show the activities of the ISTA in the next highlighting organic chemistry in Austria in a couple of years. And now let's move to Vienna. Two more universities there, or unfortunately, I forgot to tell you, BOKU is also in Vienna. At the University of Vienna, we have the Institute of Organic Chemistry, and there Nuno Maulide and our today's speaker, Davide Bonifazi, as two representatives, and then also the groups of Lothar Brecker, Roman Lichtenecker, Katarina Palic, and Matteo Lopez. And finally, we move to TU Wien, which is actually really the most eastern university by 1 kilometer as compared to Uni Wien. And here the Institute of Applied Synthetic Chemistry takes care of organic chemistry and consists of 5 research groups involved in organic synthesis. And I'll start with the group of Katharina Schröder, which will actually be the very first speaker today. And then the groups of Hannes Mikula, kind of a conglomerate of a big group between Florian Rudolph, Marko Milovijovic, myself, and Christian Stanetti. Then we have a research group of Robert Liska, Peter Gärtner, our president of the Austrian Chemical Society, and Professor Karl Kirchner. And before we start with the first talk, I actually want also to introduce a conference series to you, where the next edition is due soon, where Austria was one of the founding members, and this is the Blue Danube Symposium on Heterocycles in Chemistry, which was basically founded from the countries along the Danube, as the name clearly indicates. And the next edition will be in August, actually. You see it here, very small, 25th to the 28th of August in Prague. And it would be great if I could see some of you there in the future. So with that, I actually start introducing our first speaker and make space so she can share her screen with us. And as said, Kati is from our Institute of Applied Synthetic Chemistry at TU Wien, and got her PhD degree also at the same institute, and was then postdoc with some of the leading experts in the field of green chemistry and catalysis at the Queen's University in Belfast and the Technical University of Denmark in Lyngby. And when she came back, she started her habilitation and established a new research group focused on sustainable chemistry, completed her habilitation, became associated professor in 2018, and shortly thereafter, full professor in 2020. Regarding her achievements, I guess it's the most prestigious grant she received is the ERC Consolidator grant. And quite remarkably, she started also to study medicine at the Medical University of Vienna not too long ago. But today, she is not going to tell us about her current studies, but about her activities in organic chemistry. So, Kati, the floor is yours.
Thank you, Michi, for a nice introduction. Good morning, everyone. Can you see the slide present?
Yes.
Good. Well, good morning. It's a pleasure to be here and give this talk in this organic chemistry in Austria session. For those of you who do not know me, as Michi said, I'm located in the Institute of Applied Synthetic Chemistry here at the Technical University in Vienna, with a research group dedicated to sustainable synthesis and catalysis. And it's still a rather young group, funded about 5 years ago, that's constantly growing. And over these years, a few topics have emerged, either in the field of organo and photocatalysis, but also in more technical directions. For example, in the aspects of CO2 utilization, in the development of recycling processes, but also some projects on the development of engineering fluids. And it's actually quite unusual for me to give a talk like today that is dedicated to organic chemistry. That is something I haven't done for quite a long time, because normally I'm more talking about the field of CO2 utilization and more engineering related direction. But I want to dive back into the hardcore organic chemistry, specifically the organocatalysis, that became one of our favorite research areas over the past years. And specifically, what I want to show today is the development of the concept of counter-ion enhanced asymmetric organocatalysis, with catalyst design relying either on purely steric interactions, but also bifunctional organocatalysis that combines hydrogen bond catalysis with counter-ion catalysis, and eventually also the merging of organocatalysis with transition metal catalysis, or organocatalysis with photocatalysis. And the idea, I'll start with the purely steric organocatalysis and this first endeavors to develop the field of counter-ion enhanced catalysis. And the idea for that actually dates back to a very fundamental research question and a project that was funded by the WWF a couple of years ago, where we were wondering if you consider a flexible anion, tropoisomeric flexible anion that can switch its conformation between two enantiomeric states, whether it would be possible to combine this anion with a chiral cation, and by this pairing effect force one of the two tropoisomeric forms in one preferred form and discriminate between the two enantiomers. It was a very fundamental question that we addressed at that time, also by computational means. But this was the original idea, or the original thoughts that later on led to some developments in the field of organocatalysis. So why organocatalysis? I clearly don't have to introduce the concept of organocatalysis here, nor the people that played a large role and got the award for the wonderful developments in this field. In organocatalysis, we have in general the possibility to activate small molecules, either with covalent or non-covalent interactions, for example, via enal, enamine, or iminium formation. And what we then often have is charged intermediates. For example, if we look at the second pathway here, the classical iminium activation of alpha-beta unsaturated alkyl ketones or aldehyde results in this iminium cation. And that's an option for us to study the interplay between cation and anion, and hence ion aggregation and its impact on asymmetric catalysis. And this brought us to the concept of counter-ion enhanced catalysis. The concept of counter-ion directed catalysis is already well known and well established. We do have this wonderful system, the trip acid, chiral bulky phosphoric acid, by Benjamin List, with broad applicability. That is usually a system that relies on double steric induction with a chiral cation and a chiral anion. The main source of chiral induction usually is the acid here. It's a complex system. It requires multiple steps to produce this acid, and hence it's also quite expensive. And our original idea here was to replace this complex bulky phosphoric acid with a rather smaller system that is racemic and flexible, and combine it with a simple small organic cation, and use this in a way that the anion, the achiral anion, acts as a steric shield and rather enhances the chirality developed or brought in by the chiral cation. And this is what we coined, or what we named, counter-ion enhanced catalysis. And the very first reaction that we investigated after developing this concept or this hypothesis was a simple transfer hydrogenation with Hunch ester as hydrogen donor, where we have our two puzzle pieces, one cation, here it is an amino acid as cation, and as the anion, we used several acids, either achiral acids such as TFA, or the alternative, dichiral, achiral, or racemic phosphoric acid, but also this small flexible phosphoric acid. And we were pleased to find that in comparison, we obtained the highest values, not with the double stereo differentiation, with the chiral anion, the binyl or binap derivatives, but with this flexible phosphoric acid. And all this is just a minor change of EE from 54 to 64. That's not the world. For us, it was a very important finding because it was our proof of concept that our original idea, this counter-ion enhanced catalysis, could actually work and improve sensitivities. And from that on, well, the game was on, and we started to look for alternative systems, alternative achiral but flexible phosphoric acids that could be actually synthesized very easily. For example, with thymol as very cheap nature-derived starting block that is cross-coupled and then phosphorylated, and as the cation we modified with the systems, eventually came up with this valine-derived menthol ester, and with that we had the first lead system that could then be applied in transfer hydrogenation for, well, a small variety of different starting materials, different aromatic or aliphatic substituents, and overall gave quite satisfying high yields and enantioselectivities. So this was our first encounter with, and the term of counter-ion enhanced catalysis was born for us. What we were not that happy yet about was the tunability and the applicability, because eventually this worked very nicely for this transfer hydrogenation, but we were looking for a larger platform that we can apply to many more reactions. So we took one step further and we were wondering whether we can modify the cation here of the ion pair in a way that we have the additional possibility of hydrogen bond directed amine catalysis in combination with our phosphoric acid. And the reaction that we were interested here The reaction that we were interested here in particular is the epoxidation, the tandem enamine-iminium cyclization, where we have now the opportunity for double activation, once via iminium, once via hydrogen bond activation, and additionally, as the third interaction, we have our bulky phosphoric acid that can further influence chirality or chiral induction. So, with this system in hand, we looked again at the development of the proper combination of the puzzle pieces. And if you look first at the state-of-the-art system that was published by List already, the diamine in combination with the trip acids, it's interesting that the acid here is not playing a major part and only weakly adding to the selectivity. So whether you use trifluoric acid or the complex trip acid makes a mere difference of 2 in enantioselectivity selectivity. So there's clearly something to improve and more potential for the anion, the phosphoric acid here. And our breakthrough came when we switched the phosphoric acids towards a system that has not the biphenyl system and the 7-membered ring here, but eventually an 8-membered ring. Also, in terms of amine selection, there was small screening involved, varying the tertiary amine part, but also the amino acid derived part. Eventually, we came up with our preferred system. And just to show you how much work it was involved in here, and Fabian did a fantastic job optimizing all these parameters, came also up with a simple synthesis of how to build this complex phosphoric acids in 2 steps only: condensation, then ring closure with phosphor oxychloride, hydrolysis. So we have a cheap, fast, and tunable synthesis. And the breakthrough was obtained by the development of phosphoric acid 9, which then became our favorite system that could be used with broad applicability for a number of reactions. To show you first the epoxidations that were our original target, both with aliphatic or aromatic systems, we could react a broad pool, starting from the very simple ones to the more substituted or more sterically hindered ones with overall excellent selectivities. And with the system in hand, of course, you want to look out for other alternatives that you can react. And if you do epoxidations, a next step would be to look into the direction of acerinidation. And also here, we are pleased that the same system, again, the combination of this primary, tertiary, secondary, tertiary amine in combination with the phosphoric acid with the 8-membered ring was the best system to provide a number of aziridines in high selectivities. And then the game was on, because if you have a system that works quite in a broad range for different reactions, we of course do not have to stop with epoxidations and aceridations. And the next thing that we were looking at was acer-Michael aldol reactions, aiming for the creation of more chiral centers at once, and then the formation of octahydroacrinidines. This time with different starting materials, this amino alcohol. And it was a wonderful finding here that this reaction, that so far, to the best of our knowledge, this formation of octahydroacridines was only possible with tosylated, protected amines here. It was running smoothly in a one-pot reaction under very pleasant conditions. Again, with the preferred catalyst system, PE9, phosphoric acid 9, with the 8-membered ring here, and the selected amine component in a tandem acer-Michael aldol reaction to form the octahydroacridines and 3 chiral centers in one step, and overall with excellent selectivities, and also the sole formation of one enantiomer. And now we had what we wanted. We had the generality, we had the versatility, also the tunability on the catalyst to go to a wide direction of different systems. What probably was even more fun after the tandem acer-Michael aldol reaction was the further step towards Michael-initiated ring closure reactions, where we could create or attain high selectivity with multiple stereocenters, with 5 stereocenters in one single go. And this started because originally we were looking at the formation, we were more interested in the formation of cyclopropanes with rather moderate yield. This was rather disappointing when using bromo acetone, well, moderate results. When we switched to the chloroderivative under otherwise similar conditions, we observed something else. We had the Michael addition taking place. However, by playing around with the conditions under thermodynamic conditions at 50 degrees, something completely else happened. We had a ring closure and created in one step these 5 new stereocenters towards this bridged tricyclic system. And it worked very well with both catalysts in both enantiomeric forms. We could also create the both optical antipodes, again with very high selectivities in this complex systems. And in order to, well, if we look at this more complex, rather complex reaction, what do we think is going on as well? We have starting point, the formation of the iminion that is then in a Michael reaction attacked by chloroacetone. Then we form the intermediate cyclopropane, and then we think there is the ring opening happening and an intramolecular attack and ring opening that eventually leads to our bridged 3-ring bridged system with 5 new centers. So that was the story of counter-ion enhanced catalysis, especially with the systems that have the additional possibility for hydrogen bond formation, and then became a platform strategy for multiple reactions. But there are, of course, also other possibilities to combine organocatalysis or the phosphoric acids that we have developed in some form of counter-ion catalysis. And of course, in transition metal catalysis, we can also play around with cationic transition states, cationic intermediates, often in transition metal complexes, and enhance this or bring the phosphoric acids in the game that are the overall topic of this talk today. And one strategy where we looked at this possibility to combine enamine cycles with palladium cycles, but also bring in the counter-ion effect by an appropriate phosphoric acid is shown here. It's a Trost-Tucci reaction, the direct alylation of aldehydes. Specifically, we were interested in reactions where we could work directly with the allyl alcohol rather with an allyl component that requires here a better leaving group, so that we would form only water as byproduct. What do we need in terms of components? We need an amine so that we can form the enamine cycle. We need a palladium source, and we also need an acid. And it's worth noticing that with amine and palladium source, we have some selectivity. If we change the amine from the valine tertiary ester to the symmetric cyclohexane diamine, selectivity is already much better. But of course, there is also the role of the phosphoric acid. First of all, if we run the reaction without palladium source, then we have no conversion at all. Not surprising, after all, we need to form the palladium allyl complex. But also, if we run the reaction without acid, the selectivity is much lower. So here again, we have this counter-ion enhanced effect on enantiomer selectivity, showing that a phosphoric acid, even if it is achiral, can, by steric interaction, enhance selectivity quite dramatically. And of course, without amine, we still have conversion, but of course, no enantiomeric selectivity, which also shows you that the phosphoric acid itself is achiral and that the amine is the only source of chirality here. Right. Again, we started to investigate the role of the phosphoric acid to find the matching puzzle pieces for the direct alylation of carbonyls, looking at Lewis acids with little success, also at Brønsted acidic co-catalysts that in terms of conversion worked quite nice, but in terms of selectivity, there was room for improvement, to put it mildly. But with the phosphoric acids, we were back in the game, and especially the candidate that we had before, the one that was synthesized from the cheap and nature pool derived hymal derivative. One simple step was our lead candidate here, providing the desired high selectivity and yield at the same time. So now we had the system in hand, the right combination of phosphoric acid and of amine. What was even more important in order to drive this reaction to completeness and to high selectivities and yield was the ratio of amine and phosphoric acid. And it turned out that we require a certain excess of the amine. And only then, in this ratio 8:1, we were able to obtain a high selectivity and yield at the same time. Otherwise, we would have much lower values. The reason for that was that we have a competing enol intermediate formed. And if the ratio of amine phosphoric acid is too low, we are in a competing enol pathway that would give us rather low selectivities. whereas we have to push the enamine spaces, which was only possible in higher ratios here. And with the system in hand, with the catalyst in hand, we could again take full advantage of this enamine cycle in combination with the palladium cycle, in combination with the counter-anion enhanced effect. Here's a small selection of scope that we did here, and I'm not going through all details for the sake of time. But overall, it was a nice substrate scope that gave us quite satisfying selectivities and yields for a number of aromatic or aliphatic substrates. Eventually, what we think is happening is that we have two cycles, two catalytic cycles happening here, starting with the formation of the enamine that is then bridged by the phosphoric acid to the cationic palladium allyl species. And this is then reacting to form our chiral molecule before we enter the cycle again. Ultimately, it was a reaction that could nicely show that with organocatalysis and counter-ion catalysis, that this concept of counter-ion enhanced catalysis with the three pieces of puzzle that have to fit together, it's possible to run this direct alylation with high selectivities with only water as byproduct, and with this crucial role of the phosphoric acid to bring the selectivity from moderate values up to 99%. And with that, I just have a last piece of my puzzle remaining, and for the sake of the time, I should speed also up a bit. So far, we've been working with enamines or iminium intermediate species with the option of counter-ion enhancement. But of course, we can also combine the organocatalysis not only with transition metal catalysis, but with photocatalysis, and that enables an entirely new reactions depending on whether we have this enamine happening or existing in the ground state, as it would be the case in the conventional organocatalysis, or whether by single atom electron transfer, either via the formation of an EDA complex or via direct excitation, we can have an enamine present in the excited state. What we were interested in is the development of photoorganocatalytic alpha alkylations of ketones, so that we could have an enamine cycle in combination with the photocatalytic cycle. And specifically, we were interested in the alkylation of ketones, which is the more tricky, or which is the higher hanging fruits, because the reactivity of the ketones in this photochemically induced alkylations is much lower, and you will find only few examples of ketones rather than the work on aldehyde. Our catalysts in this system, well, now we went away from the, I have to go back, we went away from the diamines derived from amino acids, but this time we stayed with the quinine system, a phenyl derivative of the quinidine. And with this, we approached this tricky alpha alkylation of the ketones, and by a number of control experiments, we could verify that this is indeed a light-triggered reaction, because in dark, we could not observe any advance any product formation. Furthermore, from new release studies, we've seen that there is no electron donor acceptor complex involved, so it's rather a direct excitation. We also saw we could prove the radical mechanism, but from quantum yield, we could also see that there is no chain mechanism involved. So what we think is happening here is that we have the light-triggered initiation and the light-triggered formation of the diethyl malonate radical that can then react with our preformed enamine. Now we have a radical chain happening. The alkylation, eventually the product is cleaved, and we recover the starting material. And this is currently ongoing work. By now, we have quite a broad substrate pool with good to moderate yields, with selectivities. Well, there's still some room for improvement, but what we're also doing these days is we're transferring photocatalytic alkylation into continuous flow, so that we can circumvent the limitations that we have in small scale. And this is working quite well. Under optimized conditions, we can maintain selectivities of 84%. And this is currently running in my group. And I hope that a lot more reactions will be performed like this in future. And with that, I'm at the end of my talk. I've shown you different examples, starting from this very fundamental idea of how to pair or how to investigate ion pairing effects, and from a very fundamental level towards and how to use ion aggregation for the induction of chirality towards the application in organocatalysis. First with purely steric parameters. This is where the concept of counter-ion enhanced organocatalysis was born. Later on, towards this dual activation, hydrogen bond and counter-ion enhanced chiral induction, towards the merging with transition metal chemistry and latest organocatalysis. And with this, what's left for me is to say thank you to my students, to a fantastic team. It is especially three names that I want to mention here: Adam and Fabian, my dream team in the field of organocatalysis that started this field from scratch at a time where I was basically not working in organic chemistry anymore. And I'm very grateful that their hard work and their dedication opened this field of organocatalysis and of organocatalysis for our group again. Also for Michi Weiser, who was involved in bridging the organocatalysis with photocatalysis. Finally, funding from FWF and from the ERC. And finally, I would like to thank you for your attention.
Great. Congratulations, Katrina. To use your own phrase, the game is on. Well done. That's a great start to the session today, and it's now open for questions from the panelists, who may just raise their hand and ask the question. But for those who are attending, if you wish to ask a question, you can put it into the questions and answer box on the Zoom, or you can ask your question, raise your hand, and I will unmute you. So Rolf, I see you've already raised your hand, so you're
yeah. First. Thank you, Katerina. Congratulations. So If I would have been asked to if this concept will work, I would be skeptical. So I think it's fascinating that you can use now a really inexpensive source of chirality to get this very high selectivities. Katharina, so Ben List also not only pioneered this chiral phosphoric acids, but also sort of this sulfonamide catalyst, co-catalyst. Did it work in your case? Did you ever sort of try them?
We did synthesize them. And also investigated the sulfone imides because they have even higher acidity. In these reactions that I've shown here, they were not in any way beneficial. But we have some other alternatives, some other reactions currently in development where we think that the super acidic sulfone amides or sulfone imides will be of use, but that's something, well, you still need to wait a bit until I can show you some good applications for the systems. Yeah,
Thank you.
it turned out because the original idea that I had in this FWF project that the chiral cation can make the flexible anion also chiral, that was actually wrong, and we could never prove it. It still turned out that this combination of the phosphoric acid, the achiral one, with cheap chiral cations can be extremely useful for catalysis, and something good to learn that even if your original ideas are faulty, you can still make exciting chemistry with it nevertheless.
Absolutely.
Great, thanks, Rolf. So, Katharina, I have a question myself. In the area you discussed where you merged the organocatalysis with transition metal catalysis and the Suji trostylylation, do you have any idea which matched pair is responsible for the sense of enantioselectivity that you get?
Let me just go back. In terms of matched pair.
Yeah, I mean, your phosphoric acid is epimerizing. Is it?
No, not that species. So that species is simply the rotation barrier of that specific phosphoric acid 5 is already too high to be epimerizing. So here we really have the mixture of the two, the racemic mixture of the two enantiomers in the reaction.
Okay, but is there a preference then with your chiral amine?
There must be, but I can't tell you
Okay.
which one would be the preferred one. I think it would be a good idea to study this with, we try to separate the two enantiomers preparatively, also via crystallization, but we always failed to isolate them because that would have allowed us to study this, and that is something we could not do. I think it would be worth going into computational studies here to see whether we can identify which of the two acids is the one that's responsible.
Perfect. Thanks, Katharina. Now, I don't see any of the questions coming in on the Q&A box, and perhaps in the interest of time, we might move on. So again, Katharina, the game is on. Well done. We're off to a good start.
Thank you very much.
Okay, back to you, Michael.
No, I will take over now, Bet.
Oh, sorry. Apologies. Mario, over to you.
Yeah, so welcome everybody. My name is Mario Vasov, and as Michael said before, I'm now heading the organic chemistry work group since November last year. And so it's my pleasure now to introduce the next speakers. And our next speaker in this lineup will be Fabian Prengle from the BOKU, the University of Natural Resources and Life Science. And actually, Fabian studied chemistry in Germany, where he got his PhD from Professor Reisig's group in 2010 in Berlin. And afterwards, he moved to the US to the Scripps Research Institute for a postdoc with Professor Paulsen before going back to Germany, again to the Berlin area. And he started his independent career there as a group leader at the Max Planck Institute of Colloids and Interfaces. And he also got his habilitation there in 2019. And then in 2020, he accepted a call to chair the Institute of Organic Chemistry at the BOKU. So this is when he came to Austria. And he started a very productive program there, centering mainly around carbohydrate chemistry. And I also want to very briefly mention that his research was, I think, last year awarded with an ESC Consolidator grant. And his talk today will be especially about glycans. So, Fabian, the screen is yours.
Yeah, thank you very much for the introduction and also, of course, for the opportunity to participate today. This is a very nice opportunity and yeah, to see other organic chemistry happening in Austria. Yeah, let me say at the outset that we do very interdisciplinary work, actually, including also biochemistry and genetics. And our motivation always comes from the desire to answer somehow biological questions. But our main daily business in the lab is actually maybe about 80% pure synthetic organic chemistry. And yeah, as mentioned, my group is interested in glycans and particularly in plant glycans, more recently also in algal glycans. That's what the ERC grant is about. And those glycans are relatively underrepresented in the carbohydrate chemistry field. Plant glycans are actually quite fascinating molecules. Somehow nature managed to generate a very complex material sort of that allows plants, such as these redwood trees here, to grow up to 100 meters in height, and some of them are up to 2,000 years old. And the major component of this material are glycans, of course. And what we would like to do is somehow contribute to figuring out their structures, contribute to figuring out how nature synthesizes them, and also how these glycans are important in plant immunity. And in this nice cartoon here, you can see a plant cell with a cytosol in the center. And now in a growing cell, first a primary cell wall is laid down and then a secondary cell wall. And the secondary cell wall is only found in specialized cells, such as in wood and other materials. And the major components are always these cellulose microfibrils, and they're interconnected by these complex and heterogeneous hemicellulose polysaccharides. In the primary cell wall, we then also have pectin. This is acidic polysaccharides. And in the secondary cell wall, we have lignin that is also sometimes covalently bound to hemicellulose. And this all gives this composite network that is so important for these properties of the plants. Here are the polysaccharides shown that we are most interested in. The hemicelluloses include, for example, xyloglucan, glucuronoarabinoxylan, then galacto and galactoglucomannan. And then the pectin is thought to consist of different domains: homogalacturonan, rhamnogalacturonan 1, and rhamnogalacturonan 2. And we are actually synthetically interested in all of these glycan classes. And if you're not familiar with the symbol nomenclature, every little funny circle or star is representing one monosaccharide, for example, glucose, mannose, galactose, xylose, and so on and so forth. And the research directions we are interested in is the synthesis of oligosaccharides related to plants and algae, as I was mentioning, and we're using, besides traditional solution phase chemistry, also automated glycan assembly and also some enzymatic methods. And then we try to apply the synthetic structures in either plant biology or now, starting soon, in marine biology, That's referring, of course, to the structure from algae. So a typical example for synthetic efforts would be, for example, oligosaccharides related to the pectic polysaccharide rhamnogalacturonan 2. And this is a highly complex polysaccharide, actually the most complex that exists, structurally complex that exists in nature. It contains 12 different monosaccharides, shown here in different colors, and they are connected by 20 different glycosidic linkages. And these molecules are quite special. Two RG2 molecules, they like to form these borate diesters, but they only form them between these APOs moieties here, only of the side chain A of these RG2 molecules. There's also APOs in side chain B, and there's no borate diester observed. And this dimerization is extremely important, as for example, if boron is missing or deficient in the plant, then they don't grow well. So these bridges are essential for the plant. And synthetic oligosaccharides, such as here the side chain A oligosaccharide, they are very valuable compounds for further studies. So that's why we wanted to prepare this side chain A shown here, and we devised now by the time a couple of different retrosynthetic analyses, and that's the current one. So of course, first you have to design a protecting group pattern for the final oligosaccharide. That's typical protecting groups such as benzyl groups, benzoyl groups, methyl esters, here also acetonides. And then we set the first cut here to make a 4+5 glycosylation to be as convergent as possible in the synthesis. And then we have a tetrasaccharide donor and a pentasaccharide acceptor, and those then need to be synthesized again. And I will spare you the details out of these 9 monosaccharide building blocks. So Uwe Oswald in the group about 5 years ago set out to synthesize this molecule, and of course we skip most of the synthesis, and I will jump in by the last steps of the synthesis of the pentasaccharide acceptor. So we have here a galactose and the APOs, and this disaccharide is glycosylated with a trisaccharide donor. That's a thioglycoside, so the thiol here is activated with a thiophilic reagent, and then we can couple this disaccharide with this trisaccharide. Afterwards, we notice that we need to convert at this stage of the synthesis these primary hydroxy groups of the galactoses into the galacturonic acids that are needed later. So the TBS groups 1, 2, 3 here, they are removed using ammonium fluoride, and the resulting primary hydroxy groups, they are oxidized to the corresponding carboxylic acids, and then here methyl esterified using TMS diazo methane. And then the final step left is to remove the PMB group with DDQ to get the pentasaccharide acceptor. And this one actually carries a linker with a NBN-ZBZ group, and we always like to have this linker for further functionalization later. The tetrasaccharide donor is synthesized starting from this disaccharide. We notice that the thioglycoside here is 2 label at the anomeric position, so we exchange this for a paramethoxyphenyl group, shown here. Then the acetyl group is removed by methanolysis, and the resulting hydroxy group is glycosylated with this glucuronic acid donor. That was a quite tricky reaction, but Uwe sort of managed and got 46% yield in the end. Afterwards, the DMPA group is reductively cleaved using zinc, and the resulting hydroxy group is then glycosylated with L-galactose to give this tetrasaccharide. Here now the OPMB group is cleaved using cerium ammonium nitrate, and ABZ group is installed. That's this orthoalkanyl donor that can be activated by gold catalysis. And we chose this donor because it allows glycosylation at room temperature. That is good for this very unreactive acceptor here, and it's also quite neutral, so we don't lose these acetonide protecting groups. And just very recently, he now really succeeded in coupling these two oligosaccharides to give the nonasaccharide, and we are very happy about this. So hopefully also the remaining steps, the deprotection reactions, will work, and we can then have this project completed after 5 years now. Okay, so other oligosaccharide synthesis projects going on are shown here. Kati, for example, is synthesizing side chain B of the very same RG2 polysaccharide. Remya is making some oligosaccharides related to rhamnogalacturonan 1. Then we have Nivedia making some galactomannan oligosaccharides, and Zara made some Croydon oligosaccharides that are related to brown algae. So this is all done by traditional solution phase chemistry, but in cases of more repetitive structures with no exceptionally difficult glycosidic bond, also automated solid phase synthesis, also called automated glycan assembly, can really speed up this process. And this technology was developed now more than 20 years ago by Peter Seeberger, and I have used this method extensively during my time as a group leader in his department at the Max Planck Institute in Golm in Germany. And in automated glycan assembly, protected monosaccharide building blocks, like here, they are sequentially added to a linker functionalized Merrifield resin, always with a deprotection step in between. So like here, FMOC is cleaved under basic conditions before the next building block is added, or NAP is cleaved under oxidative conditions before the next building block is added, and so on and so forth. And then after cleavage of the linker and global deprotection, we can get oligosaccharides such as this arabinoxylan oligosaccharide that we have made many years ago back in Golm. And previously at the MPI, I've used this home-built synthesizer, and now since very recently, since a couple of months, we have also our very own commercial synthesizer here at BOKU now, and are heavily using it now. AGA is particularly useful if you want to make collections of oligosaccharides from just a few building blocks. So we made all these oligosaccharides, arabinoxylan oligosaccharides, just from a small number of building blocks, and had them then available for our biosynthesis studies. And here is shown the more or less full collection of oligosaccharides we have made by AGA so far. It's of course expanding, and I have to update this slide sometime, but they are all fragments of plant cell wall polysaccharides. Yeah, as I was mentioning, we also try to use some enzymatic methods for the synthesis of such structures. And a while ago, I came across this publication from the group of Yuval Shoham, where they used the glycosyntase enzyme to polymerize these disaccharide fluorides from xylan. And the glycosyntase is a mutated glycosyl hydrolase, where the catalytic nucleophile in the active site has been exchanged by a non-catalytic residue, such as alanine or glycine. And then it's absolutely inactive in hydrolyzing these products, but they can still bind the substrates. And if you have a leaving group here, such as this fluoride, they catalyze the reaction irreversibly. And so what they could make was like make very large polysaccharides through polymerization of this disaccharides. So what we thought is, can we also convert this method in a stepwise synthesis? And there was also precedence in the literature that this is possible. So we put a THP protecting group here in the terminal position, made the fluoride, and then you can do a one-to-one coupling with such a disaccharide to give the tetrasaccharide. And that can then in turn be converted into the fluoride again in a few steps. And then you have the tetrasaccharide fluoride, and you couple it with a tetrasaccharide acceptor that also can easily be obtained here by just acidic hydrolysis of the THP group. And those two together give an octasaccharide, where we remove the THP group, and this octasaccharide can then be coupled with the very same tetrasaccharide donor using the glycosyntase, and we reached the dodecasaccharide stage. Unfortunately, we couldn't go longer, although I originally wanted to. But yeah, you very quickly also with this method run out of material, and you have solubility issues. But it's a nice method to also generate very large oligosaccharides as an alternative to purely chemical synthesis. Okay, so pretty much all of our compounds we print as glycan microarrays for further studies. And this is the version that we made like a couple of years ago. There's all the glycans you have shown before, a few more, and also some glycans from collaborators. And these are printed on these glass slides where you can print really picoliter amounts, and you can print thousands of structures on these little slides and then incubate with different glycan binding proteins and so on. And we use this glycan microarrays for different applications, for example, for the characterization of cell wall glycan-directed antibodies, for the characterization of glycan binding plant immune receptors, and we also developed an assay where we can look at plant cell wall biosynthetic glycosyl transferases, so the enzymes elongating the glycan chains in the plant. And I would like to talk a bit about the last example. So how are these polysaccharides actually synthesized in the plant? These cell wall polysaccharides, such as hemicelluloses and pectins, they are synthesized in the Golgi apparatus, and then they are transported via vesicles to the cell surface, where they are then incorporated into the cell wall. And there are about 120 different glycosyl transferase activities needed to produce all the linkages in the cell wall. But of course, there are many more enzymes due to redundancy and multiple GTs with the same specificity. And today, only 24 or a few more of these activities have been characterized. So there's really a big open space that needs to be filled. And what we wanted to do is to kind of generate a more high throughput assay to look at these and characterize these plant cell wall biosynthetic glycosyl transferases. So what we wanted to do is to incubate our array with proteins with potential glycosyl transferase activity together with these azido-modified donors. And the enzymes we either express ourselves in HEC293 cell lines, or we obtain them from collaborators. And once the glycan array is incubated with a donor and enzyme, the azido-modified donor will be incorporated, attached to certain acceptors on the array, the ones accepted by the enzyme, and afterwards we can click on an alkynyl functionalized dye to then see which acceptors have been accepted. And Irene Passini and earlier students in the lab have synthesized this collection of different azido sugar nucleotide donors that we have now available to screen plant GTs on this array platform. And just as an example for particularly tricky synthesis, I show here the synthesis of arabinofuranozole donor. So first the azide was introduced by simple nucleophilic displacement here of the mesylate. Then afterwards the anomeric acetate is hydrolyzed, And then the tricky steps start. So since we cannot make use of any directing group effects, we just reacted here the free anomeric group with diallyl-chlorophosphate. And we're lucky that the undesired alpha anomer here in the product, that was not stable on silica gel and decomposed, and we could isolate the pure beta compound in 31%, which is actually a good yield for these type of reactions. And after allyl protection here, we then coupled the resulting sugar 1-phosphate with UMP methylidate to give the final donor after removal of the acetate protecting groups. And this is very tricky because these molecules, they tend to form here cyclic phosphates because they are 1,2-cis. So you have to work in buffer all the time for purification, for NMR analysis, and so on. But we could manage for all these sugar nucleotide donors. Of course, there are more, and we try to make more in the future. So there's one class of glycans we're particularly interested in. These are these arabinogalactan proteins, or AGPs. They have beta-1,3 linked galactan backbones with beta-1,6 linked galactan side chains. And they can also carry other monosaccharides. And the fucoses shown here with the red triangle, they are added, for example, by this fucosyltransferases FUT4 and FUT6. And you can see how important these little fucoses are for the plant, because if you knock out, make mutant plants that do not have FUT4 and FUT6, the growth of the plant is severely impacted, and they are really dwarfed. So even if this is only a small modification, it's important for the plant. So we used this fucosyltransferases as a proof of principle, and Colin Ruprecht in the group, who is a plant biologist, he incubated the glycan array with azido GDP fucose and FUT4 or FUT6, and you can see that the donor is really incorporated into specific glycans on the array, and it is exactly the expected oligosaccharides carrying an arabinose alpha-1,3 linked to galactose. So here, so it makes perfect sense. We then also looked at galactosyltransferases. There was published a galactosyltransferase that is supposed to make this 1,6 galactan side chains, and then we found that actually only the 1,3 linked acceptors served as acceptors on the array. And after further studies, we could actually figure out this way that it's not the right activity that was published, but they are rather 1,3. They rather make this beta-1,3 linked galactan backbone. And that we could figure out quite quickly using this glycan array. Colin then also expressed previously a completely unknown gene and called it GALT10. And here he found that this one actually used the 1,6 linked acceptors and not the 1,3 linked acceptors. And we thought maybe now we have found this activity for making the 1,6 linked side chains. But then he did some HPLC assays where he used this substrate and added 1 or 2 galactoses using this GALT10 enzyme. And he noticed that the products have a totally different retention time than the standard that we already had made chemically. So this was confusing. And then he took larger amounts of this disaccharide, added 1 and 2 galactoses, and could isolate these products. And then Markus Blaukopf, he could confirm by NMR analysis that these structures are formed. So we have a 1,3 linked galactose attached to the 1,6 linked disaccharide. And this was puzzling because somehow this structure was not reported to exist, the structural element. So what we believe is that maybe this backbone makes these little kinks and they have not been observed so much. So together with Johannes Helm and Johannes Stademann from BOKU, who are experts in glycan mass spectrometry and LCMS of glycans, we analyzed some plants. So these are tobacco plants. The AGPs are isolated, and then the polysaccharides are chopped into trisaccharides, and then we could have a look at the little fragments, and we could observe a little bit of this structural element. And then Colin, he made overexpression lines in tobacco where these plants are infiltrated with the plasmid, encoding this GALT10, and he could really observe that then a bit larger but reproducible amounts of this little kink structure or element are really formed. So we believe this is the natural function. And we have another postdoc, Duha. He made mutants missing this GALT10 in Arabidopsis by CRISPR-Cas technology. And next will be to analyze these mutants, how the pattern of trisaccharides has there changed in these mutants. Yeah, this is a little bit an idea how we try to use the synthetic compounds to then really investigate biosynthesis. Here's just a very quick last example. I don't have to go in detail. I just wanted to show that we can also use azido-modified UDP rhamnose, and we found another enzyme additionally to the published enzymes that can elongate the backbone of rhamnogalacturonan 1, adding here further rhamnoses to the backbone. Okay. So I think my time is up, and I skip a conclusion slide in the interest of time. I would like to thank all the people in the group. You have seen the pictures and the names on the slides. So these are all the people doing oligosaccharide synthesis in the group. Then there are 2 postdocs who have no training in synthetic chemistry. Duha and Niels, they do enzyme expression and plant biology. I'm extremely thankful to Markus and Colin, who really keep the whole lab running together with Thomas, and they also have their own FWF projects and they lead their own research. I thank the collaborators, then all the funding agencies from Austria and also still from the past in Germany. And of course, I thank you very much for your attention.
Super, Fabian. Excellent presentation. Complex synthesis of complex carbohydrates. Super stuff. So again, we're open for questions from panelists, but also from the attendees. And again, to remind the attendees, you can put your question into the Q&A, or you can simply raise your hand and I will unmute you. You can ask the question yourself. Rolf, off you go again.
Fabian, you showed us the synthesis of this acidic sugar and a nucleotide, and you mentioned that it's the beta anomer, but I thought how it was drawn was the alpha anomer, the dashed line.
Do I mix up alpha and beta nomenclature sometimes? Well, with this furanosis, it's always difficult on the first glimpse. I will check this if I just made a little boo-boo here and were misnaming it.
Yeah, but it could easily be I'm wrong. I'm not a carbohydrate expert.
Yeah, it could be also.
Yeah, but I found it really stunning, sort of how you sort of made this complexity of these compounds. You showed us also sort of this AGA synthesizer of Peter Seeberger, and you end up with this amino pentyl oxy linkage, which I think is quite in your favor when you make the glycans arrays. However, can you have a linker which you can use for integrating when this synthesize a synthesized fragment and making it as a donor, where you could elongate it with acceptor molecules.
Yes, so there are examples also from the Seeberger group. There's also other linkers where upon cleavage you make the free reducing end, and then you can convert this into a fluoride, for example. This is possible, and they have done it, but of course the amounts you make by this technology are rather limited. So you want to make as little reactions as possible after you finish your automated synthesis. But I mean, it is possible, it has been done, but I don't think this is something that will be done on a regular basis.
May I ask, how expensive is this commercial synthesizer?
I think right now it's about 200,000. But it's only because so few are made, otherwise it's no rocket science, the whole machine.
And you know, Fabian, rocket scientists always come out well out of that kind of comment. But once you've understood the equation for escape velocity, I don't think there's much else going on in rocket science.
Yeah.
Just to sort of put that out there. Great. Any other questions? I don't see any coming through. Maybe in the interest of time, we might pause there. Again, thanks very much, Fabian. Super addition to today's symposium. Well done.
Thank you.
Yeah, then I take over again for a minute. So thanks a lot, Fabian. And we are now moving from Vienna to Graz for the next two speakers. And we're going to start with Rolf Breinbauer from the Technical University of Graz. And again, a very, very short introduction to his CV. So Rolf studied chemistry at the Technical University in Vienna, but already during his master thesis, he went to Germany, Heidelberg, and then later on he did his PhD thesis in the group of Manfred Reetz at the Max Planck Institute in Mülheim. After this PhD thesis, he moved to Harvard working with Eric Jacobson, and then in 1998 he started 1998 I think he started his independent career in Dortmund at the Max Planck Institute, and he spent some time then as a junior professor in Dortmund before accepting his first W2 professorship in Leipzig. And since 2007, he holds a chair for organic chemistry at the Technical University in Graz. And at this time, he really established a very diverse research program looking on medical chemistry, but also palladium chemistry, which will be the topic of today's talk. And before I hand over the screen to Rolf, just one more sentence. Rolf is really one of the person who contributes a lot to our society. So he has been in a lot of panels, FWF, Austrian Academy of Science, Monotherapy for Chemie, and so on. So he really serves our community very, very enthusiastically over all these years. And with this, Rolf, thanks for accepting the invitation for today's talk. The screen is yours.
Thank you very much, Mario, and special thanks also to Pat and Michi for setting up this symposium. So it's very privileged to be among this audience and speakers. So it's also today a sort of a personal audience, because as Mario mentioned, I was trained in catalysis and then got fascinated by opportunities chemical biology offers for organic chemists. And I want to just sort of remind you sort of the general way sort of how a cell is organized, that our genetic information is encoded in the DNA molecule, which was 1 millimeter long, and short fragments of this are translated into a messenger RNA and ultimately translated into proteins. Proteins are again those kind of entities which carry the function which are most relevant for the living of a cell. They embody catalysis in the form of enzymes. They transport molecules or ions in and out from the cell by using ion channels or transporters, but also they mediate signal transduction or are responsible for the structure in the forms of tubulins. So whenever there's a disease problem, it's usually that a protein sort of goes wrong. And people try sort of to understand a functioning of a cell by interfering with the flow of information. And we have already heard in Fabian's talk that CRISPR is a technology which allows sort of to manipulate the genetic information. And also on the mRNA level, you can interfere with RNA interference. However, it would be ideal if we do control about proteins already with small molecules on the level where the proteins are produced, because CRISPR is a technique which is an on-off event. Proteins are sort of expressed in certain amounts over time. Some are existing, some have vanished, and therefore the direct intervention would be possible. And this is something which is very natural, because most sort of biological events are mediated by small molecules. And if you smell the scent of a rose, it's because geraniol adds to a receptor where you have sort of GPCR proteins, which are signaling is induced by the binding of geraniol. A special relationship between a baby and her mother is mediated by a hormone called oxytocin. So if you have a problem that other people check your tax return and you get sort of red-faced, it's because adrenaline is vasoconstrictive. And if you're in a situation under stress in exam, cortisol expressed as a hormone. So therefore, there's a lot of literature precedents in our cell that small molecules interfere with proteins. And you have an advantage because with this event or interference mechanism, you can do it dose dependent, so it's not an on-off event, but you have control in terms of amount of interaction, and also can make sort of time control, time dependently. So what we are aiming for is making us key, with which allows us to unlock or lock protein function. And this is something which has a certain challenge because we do it on a molecular level. However, there's a second challenge, is because we have 100,000 of proteins in a cell. And we want to achieve sort of just a single molecule which unlocks one of a specific key lock here of the many which we have in our cell. And over the years, my group has been quite active in producing these types of inhibitors, and especially in the field of lipase inhibitors. We had some success over the years. And this now brings us to the palladium, because this molecule agdistatine, which we published in 2013, turns out to be a quite useful compound, sort of to control the activity of murin ATGL. It's adipose triglyceride lipase, and we have sort of assembled it by the Suzuki coupling. And for the human ATGL, we came up with a second molecule. It's called NG4097. And we have, over the years, built up now a library of 1,000 compounds. And whenever we screen for a lipase, like for one just recently for PNTLR4, we have usually sort of quite good success finding molecules which are initially active and can be modified to this selective inhibitor of this new lipase here. So this was a story where we had quite success in using Suzuki coupling in making inhibitor compounds for certain lipases. But I want to start sort of my talk with a new topic, and it's called bioconjugation. It's the modification of biopolymers. And it goes along the direction which was shown in the previous talk by Fabian. However, sort of our molecules are also not as complex as his. So I've shown you before sort of this flow of information translating into proteins. However, there's another level of complexity, and this is post-translational modifications. So most of the proteins are actually glycosylated, hydroxylated, phosphorylated, or in certain cases they're prenylated. And as has been shown by my former boss, Herbert Waldman at the Max Planck Institute and his colleagues, for example, the RAS protein is one of the most prominent oncoproteins, is only active, it is prenylated and lipidated. So because then it has been anchored to the protein or the plasma membrane of a cell, and only in this setting, it becomes now sort of a molecular switch. And if it's mutated, it has this ongoing switch that it gives the signal to the cell that it should divide. And so during my time at Dortmund, so I got exposed to this type of research and found it quite challenging to make these molecules, sort of to study it in the biophysical and cell biology investigations. And it has been done either using prenylation enzymes or by using a total synthesis, where you make sort of these pieces of peptides and then glue them together with fragments of a RAS protein by express protein ligation. And so what would be ideal is having now sort of a ligation strategy where you use one of these 20 amino acids with a form of cysteine and have now exquisite chemoselectivity, neglecting all the other 19 other amino acids, that you have a reasonably fast kinetics, it works in water, dilute concentration, moderate temperatures. And over the years, people have established such ligation strategies. The very popular technique is the Maillé ligation, the MIG ligation, the nucleophilic cysteine adds to the Maillé. Ben Davis and Oxford have shown that you can eliminate cysteine to a dehydroalanine and then add again sort of a file nucleophile. However, you lose the information of the stereochemistry at this center. You could use the selenide reagent or this type of reagent, or this is work by Stephen Buchwald and Bob Pentelut from MIT. You can these type of reagents. Many of these reagents introduce now a linkage to the molecules and the proteins, which is artificial. So we have sort of their own value and stimulate a lot of research, but we were looking for the native linkage here. And an allyl ether should be possible. So that was our hope. And we devised a strategy using a reaction which already was mentioned in the talk by Katharina, the sucrose allylation. And so our plan was using, for example, allyl carbonate or acetate, a thiol nucleophile, and a palladium complex. The palladium complex would activate this allyl system, forming an hapto-free palladium complex. And then a thiol nucleophile could either attack at this terminal end, producing the linear chain product, which in our case would be this side one, or the branched one if it makes an internal attack. So there was precedence for this reaction in forms of palladium chemistry by Trost and Sinoué. And more recently, people like Plitka, Sao, and Carrera used iridium or iron catalyst to make the branched product. So we would be interested to using the linear product. And the challenges which we defined at the beginning of this project was that we want to have superb selectivity for the end product, because on the level of a lipid protein, there would be no change how to sort of separate the isomers. We would have to achieve a high functional group tolerance. We have to bother about catalyst poisoning. And most of all, we would like to have conditions which allows us to perform this reaction in water. So here is now sort of what we planned to do. And my first PhD student working on this project, Hilmar Schröder, screened 50 ligands. And there was just a single one, it's bifidobacterium, a ligand designed for the work in hydroformylation with rhodium, which turned out in this palladium chemistry as a superb ligand. It's cysteine selective, it gives us the preference for the N-thiol ether, and in a way it was compatible with aqueous solvent. And Thomas Schlatzer, a brilliant PhD student who continued this project, sort of investigate the scope and limitations of reactions and the mechanism. And I want to show you some of his data here. So you see sort of that it's quite a universal strategy, tolerating a wide variety of thiols and also electrophiles to form this CS carbon bond or CS heterocarbon bond. What was interesting when we observed this reaction is that it's a very fast reaction. So within 15 minutes, already have a complete conversion. However, the N-to-iso ratio is rather poor of 87 to 13. If we work a reaction after 24 hours, which we originally did, then we have sort of selectivity when we do not see any amount of a branched isomer anymore. So this was indicating that we have a level of reversibility in this transformation. And Thomas made a lot of experiments where you use either, for example, this branched product, exposing it to the palladium bifidobacterium system, and you convert it to the linear one. And he could show that if you do not add the palladium catalyst, but also a thiol nucleophile, you don't get any sort of transformation. So it's a truly palladium catalyzed reaction. And using crossover experiments where we had sort of this phenyl methylyl ether versus the allyl paratolulyl sulfate thiol ether, or the branched version of it, we get sort of scrambling, leading us to the thermodynamic mixture of all these reagents. So it was known quite for a long time that this very first step of the palladium allylation are sort of reversible. What Thomas could show is also that the final steps are quite reversible. And in principle, we could start from a thiol ether and producing thiol ethers going back and forth. And this is following along the principle of a shuttle chemistry process which has been sort of defined by Bill Morandi. And keeping in mind that fire ethers usually higher molecular weight and less smelly than fire, this might be also sort of an attractive option if you scale up those kind of transformations. Now, so these were very small molecules. What about now peptides? And a very important test peptide for us was the cysteine tyrosine molecule, because the tyrosine has been shown by Matt Francis to be also sort of a substrate for allylic allylation. However, with our reagent combination, we could show that these type of allyl ethers can be produced using this dipeptide, that it's exclusively go to the cysteine here. We can make sort of various type of allyl reagents where we introduce bioconjugation handles that we are introducing with fluorescent tag and affinity tag, or, and I will show you later, we can use a bifunctional one, which we can use for stapling of peptides. So does it work on a more complex peptide setting? And yes, it does. And I'll show you just a few examples. And I want to raise your attention to this peptide P7. So peptide P7 has 32 amino acids and was provided by Christian Becker, who was our main collaborator and partner, peptide chemistry at the University of Vienna, with whom we had sort of a very productive collaboration. And Christian provided us with this peptide where we have all amino acids bearing a functional group. So we have threonine, serines, arginine, lysine, and so on. And by mass spectrometry, we could confirm that only the cysteine amino acid has been modified. If we introduce two cysteines, we could sort of interconnect them, and this is called stapling. And this is a concept pioneered by Greg Verden in Harvard, who used this as a way sort of to stabilize alpha helix motifs, but he used different strategy where he already provided modified amino acid, which he sort of linked together in many cases by ring closing metaphysis. But this would be a more direct way to introduce staples. And in terms of a reagent, we could sort of modify also the distance between the stapling event. Does it work on the proteins? And this was the ultimate test. And we received from Christian this UBL3 protein. It's ubiquitin-like protein. It was is post-translationally modified in vivo, and it's membrane bound. So this was an ideal test case. And here we have sort of at the terminus with cysteine, and we use now a larger amount of palladium with bifidobacterium, but we get sort of excellent yield. So it's 60% isolated yield of this ligated protein. And here you see sort of the mass spec event confirming this data. It works also for a Sharon residue, or if we have two cysteines, we can introduce two of them. So this was something which was quite impressive for us. And we could refold the proteins, and showing here that we get sort of the native structure back again. Although we were working in acetonitrile water, and under the chemical transformation, sort of the protein was denatured, but it could be refolded. It also works for another protein, heat shock protein 27. Here we introduce this alkene tag, where we can now perform then copper click chemistry, introducing the biotin label here. However, I mentioned before, sort of we use acetonitrile water 1:1. And these are conditions where most proteins would denature. It would be sort of very attractive if we can diminish the amounts of organic solvent, because then the protein structures would react under native structures. And for this, we were aiming for water-soluble ligands. And in this review article, we summarized strategies how in organometallic catalysis, water-soluble ligands have been designed or produced. One way is that we use sort of a pre-functional building block when setting up the phosphine. Most often, functional groups are introduced for making it hydrophilic by direct functionalization. The famous tris-acetylphonato phosphine is produced like that. Or that we have sort of a phosphine with a carboxylic acid tag, where we can introduce now with an amide and solubilizing. So for us, that means that we convert bifidobacterium and introducing hydrophilic groups. And from the design, we were expecting that if we replace the methoxy group, for example, by a PET chain or making solubilizing groups, this should work out. However, it didn't do like what we were hoping for. So this is the synthesis of bifidobacterium. You start from this bridging biphenol, and you introduce this chlorophosphate reagent. However, when Thomas Schlatzer, sort of already in his diploma thesis, made minor changes in this bridging biphenol, it didn't end up as a bridge, but in the side chain as a ligand here. So basically, what we tried to be using in the periphery became then the bridging entity here. And this is something which I think to the day we don't completely understand. However, it has been observed before by Bella, Byrne, and Hoffman, it's just subtle changes make these ligands. Thomas had then a brilliant idea, and he sort of proposed that we replace the tertiary butyl group, with, which is the most lipophilic one, by a water solubilized group, and a phosphonate. And he synthesized this ligand, now assisted by Mark Weber, sort of his following PhD student, where we start from biphenol, making a bromination, now introducing the ether moiety by using an Ullmann type coupling. With an effort and Todd reaction, the phenols are converted to the phosphates, and then taking advantage of its ability to make an direct metallation, we can initiate a phosphor-freeze rearrangement and then making with chlorophosphate this desired biphosphate ligand. So this is now more hydrophilic because instead of a tertiary butyl group, it has now sort of this phosphonate. And this again, now tested for this UBL system protein, now works already with a minor amount of acetonitrile. So previously it 1:1, and now it's 5 to 95%. So it's 95% water. And this is a true advantage because some proteins already on these conditions become sort of still in the folded state. However, we're aiming for to going in a pure water system, but this is something which has not been sort of completed yet, but hopefully I will be able to report this to you in the future. So I want to come back sort of to our aims to making providing molecular keys, sort of switching on and off proteins. And I've shown you the slide about the lipase inhibitor, which for certain proteins we were successful. But also we are now interested in interfering with protein-protein interaction. This is another level of controlling protein function. And for example, if Rho GTPase binds to ROCK kinase, then ROCK gets activated because it unfolds. And then sort of you have this tumor invasion, metastasis, hypertension, bronchial asthma, downstream processes. So if you would prevent by a small molecule that Rho binds to ROCK, you also would shut down this downstream processes. And this was the motivation when we started this project together with Reza Ahmadian. He was the group leader at the Max Planck Institute in Dortmund and is now a professor at the University of Düsseldorf. And Reza was the first one to close the structure between the Rho GTPase and the ROCK kinase. The ROCK kinase exists as a coil, and here you see sort of how this alpha helix interacts with the Rho GTPases. And it's typically that not all amino acid residues of alpha helix participate in the interaction, but more or less just the one which look in the same direction. It's the first, the fourth, the seventh, and the 11th amino acid. So it was Andy Hamilton Yale who proposed that you could replace an alpha helix motif, which is based on peptides, and peptides are not good drugs because they get hydrolyzed easily in our body, that you use a scaffold represented by a terphenyl structure, and you replace sort of the first and the fourth and the seventh amino acid as the side chain on this terphenyl scaffold. Here we see sort of a modeled structure for our Rho GTPase target. And here, that would be our desired structure. And it took us some time sort of to synthesize these molecules. Without going into the details, I just want to mention that this was a linear synthesis where we used quite diverse chemistry, like this Miyawa-Smith relation, Suzuki coupling, functional group transformations, another Suzuki coupling, and it ended up with a structure. And it took Reza 2 years sort of to come up with the biophysical data. And he could confirm that indeed this molecule was interacting with the Rho GTPase with a decent dissociation constant. And during this process, when we did the synthesis and also sort of waiting for the data, we sort of became aware that this event of protein-protein interaction is much more universal than we were expecting. And it was Uli Stelzl at that time at the Max Delbrück Center in Berlin who made an yeast-to-hybrid assay and identified more than 60,000 protein-protein interactions in a human cell. By looking at structures in the protein database, we realized that many of these protein-protein reactions are sort of mediated by alpha helices at the active site. And Pamjit Arora made a sort of informatic analysis showing that 60% of all PPI structures involve an alpha helix motif. So this would be a huge opportunity for organic chemists to find molecules which prevent this protein-protein interaction event. However, it requires a more efficient synthesis. And Martin Peters proposed, instead of having this steppy linear synthesis, making a modular synthesis where we use sequential Suzuki couplings, starting from core fragments where we have either iodine or triflate or bromine diazonium, and then using the same set of building blocks for the top and the bottom ring, represented by this meta-substituted boronic acids. We made some exploratory results regarding sort of these core fragments, and just looking at the yields for a certain building block, it was 1% for the bromodiazonium, and the iodotriflate convinced that it would be more convenient to go for the iodotriflates. We were aiming to the synthesis of all these 18 building blocks because the glycine and the proline are not sort of relevance for events within an alpha helix. So I just flipped sort of the slides to showing you sort of how we produce these building blocks. And for the iodotriflate, we succeeded in making all building blocks, some in a protected form. However, we failed to do the histidine. So even in a protected version, it was not possible to make a stable molecule with a triflate in the presence of an imidazole residue here. So we repeated everything and did now for the bromine iodine, but now we have completed the set of building blocks, which allows us now to assemble this alpha helix mimetics. What we needed was, again, this meta-substituted boronic acid. And here we sort of modified our design because for solubility reasons, we wanted to introduce a nitrogen here because the terphenyls itself are not stable in water or soluble in water. From the synthesis, this was my most challenging position. So any other nitrogen position would be easier to make. However, we tried to do so, and just to show you two syntheses here, it started from the 3,5-dibromopyridine, converted it with Buchwald's version of the Finkelstein reaction to the iodine one, using Knochel's turbochromia, making the side chain, removing the hydroxy group by first converting it to the chlorine by zinc acetoacid. And previously, we introduced the pinacol boronic acid ester. For the tryptophan, we had sort of to assemble the ring moiety of an indole by using Fischer-indole synthesis. And the most difficult building block, again, was the histidine. And we had 6 routes explored, and the one which was successful was the one where we used this building block, Knochel's strategy, introducing the side chain, and then we had to remove it, which was possible with the samarium iodide. But on this level, the Miyawa borylation was impossible. But if we switched the order of events, first introducing the pinacol boronic acid and then deoxygenation, we succeeded in making the histidine building blocks. So this has been published just recently, and now we can make play molecular LEGO, assembling these building blocks and making these terphenyls. So what about biologic applications? And I want to share with you sort of data which we still have not published yet, and it's protein-protein action between sort of a transcription factor called SRPP. It's sterol binding protein. Once it binds here, then this transcription factor complex of several proteins assembles, and then in the Tata box, the gene is read out and we get gene expression. So our aim is sort of to interfere with the interaction between the transcription factor, this SRPP, with the Kix domain of MET15. And our collaboration partner, Haribabu Atanari from the Harvard Medical School, sort of solved the structure using NMR techniques. And you see sort of now sort of the Kix domain, this is the one in the magenta, and the SRPP protein alpha helix structure is the one in Turkish here. And it was Martin Vareka, my PhD student, who used data given by Haribabu Atanari, designing now a molecule which looked like this, and checking it for biological data. And here are some structures produced by Martin, and they showed in the biophysical experiment very early on quite decent selectivity. However, in the cell, it was not active. And then we did some medchem tricks like converting carboxylic acid and tetrazole, and ultimately we had a prodrug, which in the vitro assay is not as strong anymore, but it's active in cells. And we now can rationalize the binding of our molecules by using some molecular docking performed by Sarah Berger and Gustav Oberndorfer. And what is important now, sort of data gained at the Harvard Medical School with RNA sequence analysis, the gene expression is shown. And you see downregulation, and it's difficult to read. Here it's called steroid biosynthesis, and this is terpenoid biosynthesis. So this is exactly sort of the genes which are controlled by our binding domain. And this means that our best inhibitor hits on target. So this is something which excites us a lot. And we are continuing this type of research because we have now a full freezer of building blocks, and we could easily make sort of any permutations of this alpha helix mimetics. With this, I want to close. I've shown you today sort of two stories where we either introduced the technique of bioconjugations, now modifying polymers or proteins by making prenylations. And I also have shown you sort of how we want to design compound libraries and tool compounds, and we've been successful in interfering with the protein-protein interactions. I want to close with a quote which was very influential. Then, being in Harvard as a postdoc, I was auditing some classes, and Andy Hamayas was teaching Chem 115. It was a fascinating class about retrosynthesis. And I remember him saying, he said, palladium will always do what you expected from it. It will find its correct oxidation state, etc. You just have to find the right ligand and the right solvent and base. And I can sort of, after 20 years working with palladium, agree with this. So ultimately, any palladium reaction we wanted to establish has worked out if we optimize the conditions. I want to thank the students, especially the students who were involved in this project. I mentioned their name before, our collaboration partners, and you for your patience and audience and for the opportunity to present our research here. Thank you very much.
Rolf, super. Again, another excellent addition to today's event. And yeah, it's true about palladium. It will do what it wants, and it's not that straightforward to just find the right ligand, the solvent, and the base for those who work in it. And my question, if that's okay, Rolf, is on your palladium catalysis, where of all the ligands you found to get that good selectivity for the linear product with the carbon-sulfur bond formation, it was biphetphos was the right one. Yeah, not one that would be readily available, I would say, in most labs. So to find that one was pretty spectacular. In order to make it water soluble, you really only gave one example where you tried to make the phosphonate. Did you ever try to make the sulfonated analog of biphetphos?
We were thinking about this, but what we realized, and this is certainly a limitation, is that the phosphate ligands are acid sensitive. So we fear that this would be sort of not compatible. Another ligand which gave not as good results, but decent one, is DPBF. Okay. And here we made sulfonated versions out of it, and also sort of ammonium versions. Yeah. But in terms of solubility, nothing beats a sulfonate, because that is really sort of the most strongly sulfonated entity. Yeah. We are still continuously working on it, and probably we give it another try, that we put a sulfonate with a protecting group at the periphery of the biphenol on the phosphate here.
Right. Okay.
But yeah. It's more difficult than we were believing.
Oh, yes, that's the other thing about even ligand chemistry. That's sometimes more difficult than everything looks easy on paper. Great. Any other questions coming through from other panelists or through the Q&A from attendees? Very quiet attendee list today. So I suppose a philosophical question then, Rolf, maybe to finish up. What's the next frontier then in transition metal catalysis in chemical biology? So what's the unsolved problems? So what really would be great to get working?
Honestly, to do it without transition metal catalysis. So having a good organic reaction, I think, would be even more advantageous, because ultimately, even palladium, it's not a very toxic metal, but somehow it might interfere. And having sort of an all-organic reaction which makes labeling efficiently, I think is certainly something which is very attractive.
Right. Okay. No, fair point. Fair point. Good. So don't seem to be any further questions. So again, Rolf, thank you very much. And I'll pass over to Mario, I think. Is that correct?
Yep.
Yeah, okay.
So yeah, thanks, Rolf, also from my side. And we now just move a few streets further from the Technical University of Graz to the Karl Franzens University in Graz. And our next speaker will be Melanie Heil, who holds a professorship there at this university. And Melanie is actually from France, where she studied chemistry in Rennes. But in 2006, I think, or 2000, is it correct, Melanie? 2006, you moved to Austria. And she went to Graz and did her PhD in the group of Kurt Faber, where she really got introduced to enzyme chemistry. And after her PhD, she then did a postdoc in Atlanta, USA, with Andy Bomarius. And after this postdoc, she went back to Graz and started her own independent career, started her working group working on different aspects of enzymatic catalysis. A few years ago, she obtained a habilitation, and she is now holding a position as an associate professor working on different aspects of biocatalysis. So Melanie, the screen is yours.
Thank you very much, Mario. I also take the chance to thank the organizers and EuChemS for allowing me here to join my colleagues from Austria. It's actually funny that we do not get the chance to meet all of us at the same place. It seems the digital space is for that reason much more convenient. So indeed, we're going to make a switch back to synthetic chemistry. However, you'll see that in my talk, I will be merging aspects of technologies that, for instance, Rolf has been mentioning. So if we look at the development of chemistry, there is no doubt that chemists as a society, as a community, has really got inspired by nature, especially in synthetic chemistry. We see that a lot of inspiration has come from biosynthetic pathway. It's actually very fascinating to see how nature has evolved to develop complex organic reactions in a very efficient manner, in a very clean manner, and all that due to the action of proteins with catalytic activities of enzymes. However, if we look now at how chemistry has turned in the 20th century, I guess many of you will agree that as the complexity of the molecule increases towards fine chemicals, pharmaceuticals, also the processes are losing their efficiency. And this is actually very well reflected in the E factor. So there is no ideal green metrics to quantify the greenness of a process. The E factor is, however, very practical in representing the amount of waste generated for a given product. And we see here that, especially in the pharma area, where you're going to make complex organic molecules with a lot of chiral centers, you're also going to produce a lot of waste. So in essence, up to the equivalent of 100 times in amount of waste, the amount of product that you're actually trying to make. There has been processes that got improved tremendously by incorporating enzymes in the synthetic pathway. And I just took here one example of such a synthesis. This is a process that allows us to make L-DOPA. So L-DOPA is a prodrug. It's used for treating...
Melanie,
yes?
can I just interrupt? From my screen, you're still on your first slide, so it hasn't moved at all.
I want to apologize. Maybe I need to stop the sharing, and I'll
maybe start again. And even though you had clicked full screen,
yes, this is very intriguing. Let me see. What do you see right now?
I see your slides in very small versions, all the way down from 1, 2, 3, 4, and your first slide is still there.
Huh.
Is that the same for everybody else?
It is, yeah.
Yeah.
This is let me try this. And now?
Yeah, now you're on full screen and you're at the DOPA slide.
Okay, perfect. Yeah, thank you very much, and really apologies for that. Well, you didn't miss out so much, I guess, because this is the slide where we're talking about chemistry. So, yeah, this is L-DOPA, essentially a prodrug that is used for treating symptoms of Parkinson's disease. And we see here that one process that is actually run on a multi-ton scale rely on an enzyme. It's a tyrosine phenolase that catalyzes actually a very complex reaction. We see that we have a catechol, pyruvate, and ammonia, and all that performed in a very stereoselective manner. The reason why it's very important to get this unnatural amino acid in a very stereoselective manner, as I told you, it's a prodrug, this compound, and what your body is going to do, it's going to metabolize it by using an enzyme to decarboxylate this very enantiomer of the molecule to generate the drug, the dopamine. So just an example to illustrate how we can utilize enzymes to perform a very intriguing chemistry. So I'm also grateful for Rolf for having already shared this, let's say, information on the central dogma of molecular biology. So essentially, you will have a unidirectional transfer of information from DNA, genetic information, all the way to the protein. So this is something that biotechnologists have been utilizing now for a couple of decades. We can in silico go back from a given any protein sequence to the DNA level, and using recombinant technology, we can then produce these proteins on demand. And I would argue that nowadays it's become very routine. And in our lab, we are chemists. We're also able to generate a wide range of diverse proteins, in essence, enzymes that we want to investigate. So that's essentially what we're doing in my lab. We have a strong interest in designing biosynthetic technologies with a focus on repurposing enzymes for non-natural reactions. We also have a strong interest in a asymmetric synthesis, so accessing molecules with biological activity. And all in all, while we develop these tools, we are focusing on, well, let's say the lowest environmental impact as possible. The tools we use, as I said, we are chemists, so we do a lot of organic synthesis to generate our substrate and reference product, but we will be utilizing biocatalysis as the essential method to develop these tools. We're also entering the rather complex for us world of genomics and bioinformatics to support us in our endeavor of making chemistry greener. So today, I'll take the chance here to share some vignettes from our current research. We have various programs dedicated to redox biocatalysis. We're also interested in novel strategies for nitrational reactions using enzymes, as well as programs studying the resistance of chemically stable lactams towards hydrolysis. Today, however, I will mostly focus on the right panel of this slide, and I would like to start by telling you a story of enzymes that I've been working with for now a really long time. These enzymes are called all yellow enzymes. They are yellow because of the coenzyme that they rely on. It's a flavin. When the flavin is oxidized, it's simply yellow. So it's actually quite convenient when you produce those enzymes. What these enzymes are doing, they are able to perform asymmetric reduction of CC double bond. So the double bond here needs to be activated by an electron withdrawing group. So here it's essential that you reduce the electron density in the double bond to be able to perform a hydride transfer. So the hydride transfer occurs after a first step of reduction of the flavin by the nicotinamide molecule. I'll tell you more about that molecule a little bit later on. And the reduced flavin is then able to perform a hydride transfer onto the beta carbon of the activated alkenes, followed by protonation from the opposite phase by a catalytic residue that turns out to be, in most of the cases, a tyrosine. So this is very well understood, and we rely for that on beautiful work by Vincent Massey a couple of decades ago. So we and others have been able to utilize these enzymes for the reduction of a variety of molecules, and you see here an example, or let's say a selection of, first of all, activating groups, as well as target products that could be obtained in a very high enantiomer purity. So why do we get this exquisite stereoselectivity in the reaction? This is essentially due to the binding of the substrate in the active site of the enzyme. We see here represented with a nitro alkene, and the sphere here represents the hydride that is transferred from the reduced flavine, while the tyrosine here will be protonating from the opposite phase. So if you have a selective binding of your substrate in the enzyme active site, you will have very high stereoselectivity because we have a trans addition of a two hydrogen, formally speaking. So a couple of years ago, as I said, we had worked a lot on various activating groups. We worked on esters and let's say lactones. And we wondered at a time where a lot of interest was put towards levulinic acid as a top based bio-based chemical, we wondered if this molecule, it's called Angelica lactone, you obtain quite easily by dehydration of levulinic acid. So we wondered whether those compounds could also be amenable to reduction. On paper, it looks very complicated because this double bond is not activated by the ester group, since we have here CH2 in between. To cut a long story short, we found out that indeed you could reduce this compound here all the way to the reduced valerolactone. And after many investigations that were necessary to explain why a non-activated double bond could be reduced, we found out that the process was actually only a formal reduction reaction. And what happened is that we had two-step cascade with a first step of non-redox isomerization. So the enzyme is able to isomerize this molecule to the alpha-beta unsaturated ester. And this molecule is now, as I showed you before, an excellent candidate for the bioreduction by the enzyme. So we postulated as well some mechanism that was based on mutation, and we could demonstrate that we have acid-base catalysis where the reaction proceeds through deprotonation of the alpha position and reprotonation here at the gamma position by this tyrosine. But you see here in this first study, the product we were making was unfortunately racemic. So this acid-base catalysis is not very surprising if you look at other enzymatic approaches for isomerization occurring in nature. And there is a variety of deprotonation, reprotonation, or protonation, deprotonation occurring in nature. A very beautiful example is the case of ketosteroid isomerase, which relies on an aspartate to perform here this proton exchange between the gamma and the alpha position. We worked also on related mechanism with these all yellow enzymes for very different substrate, a substrate that is actually activated here, where the isomerization was just shifting your double bond from the external position to the internal position through a reduced flavin. So here, the key, the same as in a polyunsaturated fatty acid isomerase, the key is to have a hydride being exchanged between the two positions. So compared to this, our platform appears very, let's say, unique. So as I told you, we were unfortunately making the racemic products. So I was starting to lose interest in this project until Marina, an excellent student from Italy, came to Graz to investigate two new all yellow enzyme homologs from different sources, one from the red algae and one from a fungal strain. Also here, trying to cut this very long story short, after being able to demonstrate these enzymes are indeed all yellow enzymes, we could also demonstrate their catalytic activity. in the isomerization of this Angelica lactone. I tend to call that the beginner's luck. Marina came along with 2 proteins, and it turns out that both were not only active, but also stereoselective and work in a stereocomplementary manner, so that we were able now to generate the 2 enantiomers of our product with very high enantiomeric excess. So of course, this attracted our interest, so we decided to have a look at the molecular level at these proteins, meaning that we needed a crystal structure. This was actually performed a couple of years ago. And to get even more insights, we performed soaking, so essentially crystal soaking with the substrate. You see here the electron density corresponding to the substrate. And for reasons that for now are still eluding my understanding, we also looked at the electronic density after 7 hours. And you see here that something changed. Essentially, within the crystals, the reaction has taken place. So the reason why this is very important for us, and this is probably easier to appreciate here at the bottom scheme, is that we now have a clear positioning of a suspicious proton donor, a tyrosine here, very well located for proton transfer on the gamma position. So here we get really a great understanding on where the stereoselectivity is actually coming from. We did a couple of mutation as well to try to pinpoint again, the reasons for the stereoselectivity. And here you will see that these 2 tyrosines here were demonstrated to be crucial in not only activity as well as stereoselectivity. So we postulated that this tyrosine 66 is acting here as a proton relay and will be necessary here to activate this tyrosine that will work as a protonation for the gamma position. For the enzyme with the opposite stereoselectivity, we also managed to get a crystal structure. However, here the soaking was not successful. So what we have is an empty binding site, enzyme binding pocket, if you will. So we could only postulate based on some acidic residues, what residue could be mutated and have an influence in the reaction. Here, I would like to draw your attention to 2 tyrosine, tyrosine 78 and tyrosine 235. And if you see here, looking at the impact of the mutation, so essentially you remove the OH of the tyrosine by substituting this residue by a phenylalanine, we could demonstrate that in one case for tyrosine 78, the enantiomeric excess of the product was going very much down. But the activity was partly conserved, while mutating this tyrosine 235, that's this residue here, you were just abolishing the activity. So here we also hypothesize the reason for the reaction, and I could just summarize that in this one scheme. For the R isomerase, we postulated that deprotonation was followed by protonation on the C phase by a tyrosine. This tyrosine was getting reactivated through protonation by a second tyrosine, and then you were getting back to your starting position, so to speak. For the S isomerase here, we have a different situation where deprotonation appears to be catalyzed by a tyrosine. So the alpha position gets deprotonated, and the reprotonation at the gamma position is occurring on the opposite phase, on the re phase of the molecule, meaning that essentially you're now able to access the opposite enantiomer. So this is all fine, but what do we do out of this reaction? So if you look at the literature, there is a beautiful work by the group of Deng, who has shown isomerization of butanolides using organocatalysis. You see the reaction conditions, let's say, call for improvement. So we thought that we could utilize our platform of performing isomerization in water and combine that with a follow-up reduction. Remember, I told you the intermediate compound following isomerizations or excellent substrate for reduction. So the idea was here to develop a formal reduction of the Angelica lactone to the stereocomplementary enantiomer of the of the product. Excuse me. So here I will also have to condense a lot of research. It was very challenging to identify enzymes that allows you to have exquisite stereoselectivity in the isomerization, as well as a great activity in the reduction. And the way we solved that was to design a bimolecular protein by fusing two old yellow enzymes. One that would be responsible for the isomerization, and one that would, in a second step, be able to reduce the alpha-beta enantiomer. This is again something not difficult at all. Technologically speaking, you simply need to paste your two sequences corresponding to the genes coding to your protein, fuse them with a little linker. So this is the linker we picked here. It's a 9 amino acid linker. And then you just perform your overexpression in your favorite host, in that case, E. coli. So again, here, a fusion protein, very well active. To gain access to the opposite enantiomer of the fully reduced product here, I would say that, well, simplicity is the ultimate sophistication. So it turns out that our isomerase was also a great reductase and was able to perform the reaction all the way to the final compound. You see, however, that despite quite satisfying enantiomeric excess of the product, the conversion was still calling for improvement. So that's where we decided to screen the protein diversity, all yellow enzymes, or very diverse class of enzymes. You see here all the candidates that we have picked. This is a phylogenetic tree that tells you as well that they are clustered in different groups. And while at first you might have the impression that the stereoselectivity or the absence thereof is connected to the origin of the protein, well, it turns out that the more you screen, the more you find out this is not true at all. So what we did is that we tested all these different proteins and could identify other selective as well as non-selective biocatalysts here. So why did we do that at all? As I told you, we were trying to improve the cascade, and this turned out to be actually very successful. To obtain the R-valerolactone, one protein revealed excellent stereoselectivity as well as reductase activity. So we could obtain essentially these two compounds with better, let's say, metrics compared to before. To finish on this project, we are interested in broadening the substrate scope of this platform. And Federico here is putting a lot of efforts into synthesizing a range of butanolides. So we are currently focusing on gamma-saturated compounds, and the first results are very promising. We've been able to generate the corresponding reduced lactones you see either with a butyl or a benzyl at the gamma position, with very high EE value in a stereocomplementary manner, again, capitalizing on the protein diversity that we have at hand in our lab. And again, the cascade appears to be functioning very well in both cases, with one enzyme completing the two steps. So what are going to be the next steps? We are going to, increase the diversity of the compounds by looking at dye-substituted lactones. So those starts to become challenging for various reasons. So first of all, you will need to have a catalyst that is able to perform asymmetric isomerization on this material, generate gamma chiral unsaturated lactones, and then perform asymmetric reduction. We now have a pro-chiral moiety here to essentially achieve diastereoselective formal reduction of these butanolides. Again, here we have promising results on a couple of substrates, and I hope we'll be able to share this data very soon. So in the remaining time, I would like to switch gear and focus on redox biocatalysis by going back to that molecule I started to talk about earlier on, nicotinamide. So in biology, the nicotinamide molecule is, let's say, the favorite redox partner that you could find due to a very low redox potential here. So this has been used also in a variety of biocatalytic processes. The only caveat here is the price is very expensive. So if you want to use it in a stoichiometric amount, you're going to be limited to small scale. Fortunately, decades of research in this area have provided us with alternatives. We call that regeneration strategies that allows you to essentially couple your targeted reaction of interest, let's say a reduction, to an oxidation that will consume a cheap co-substrate and generate a co-product. It could be, for instance, glucose that will be oxidized to gluconolactone through the action of an enzyme, glucose dehydrogenase, that relies on the oxidized form of nicotinamide. And there are other options. Again, here you will need a second enzyme and you generate stoichiometric co-product. Other options are available if your enzyme of interest can function in, let's say, two different redox directions. So that your enzyme would be able to reduce the substrate, generate oxidized nicotinamide, and at the same time, it will oxidize a co-substrate, thereby reducing the cofactors. So that's called the coupled substrate approach. Again, here, although it's simplified because you only have one enzyme, you will rely or you will end up generating waste in form of a co-product. So a couple of years ago, we wondered whether we could design a simplified system that would essentially lead to performing redox chemistry in a redox neutral manner. So what we thought here is how about having internal shuffling of electron, combining at least two reactions within one substrate to generate a single product and no co-product here. So the idea that we started to develop was to work on let's call them bifunctional substrate, again, a molecule having two handles that you could oxidize or reduce. So essentially redox chemistry in different directions. So to kind of make it clear, I will share with you the model substrate that we started working with. So the enzyme was an alcohol dehydrogenase. It's an enzyme that is known to catalyze reduction and oxidation. So we thought, well, if we could have a first step of reduction of this aldehyde moiety, the bottom one here, generate corresponding alcohol. We generate a molecule that will spontaneously cyclizes to the corresponding lactol. And lactol happens to be excellent molecule to be oxidized by the same enzyme so that we would generate a lactone. All in all, you would only consume catalytic amount of nicotinamide. So actually, essentially, you don't consume it at all. You just regenerate it while the system is working in this, let's say, 1,4 hydride transfer. So it turns out to be working very well. And we've been able to apply this technology to a range of dialdehyde. You see here working on symmetric aldehyde to non-symmetric one, here also relying on the regioselectivity of the enzyme. So essentially here, as you increase the chain or the linker between your aldehyde and your phenyl ring, you are able to rely on a preferential reduction of the bottom aldehyde to generate essentially this one regioisomer of the lactone. We have also increased the bulk of these compounds by working on biphenyl dicarbaldehydes. You see here that the amount of nicotinamide was very low. Again, it's catalytic since we have this efficient internal recycling of the nicotinamide. And the proof of applicability was shown on a range of dibenzoxypinone products. You see here a sample of the compounds we were able to generate. And without going into the detail, I would simply like to highlight that depending on the electron property of your substituents, you could also influence the regioselectivity of the reaction. And this is shown here by having the fluorinated substrate here, derivative, on the side of the reduced aldehyde, while the electron donating group was favoring oxidation. And finally here, we decided to couple the synthesis of this bi-aryl substrate that you obtain simply by Suzuki coupling. By that, we were inspired by pioneering work by the group of Harald Gröger, who showed the compatibility of palladium chemistry with alcohol dehydrogenase. So we were able to do that cascade of Suzuki coupling with disproportionation of the dialdehyde to the lactone in a nice one-pot cascade. So to finish for today, I would like to share 3 more slides roughly on how we are currently utilizing this redox neutral system to make lactones and move towards, let's say, maybe unusual scaffold, at least in biocatalysis. And for that, I'm also grateful for Katharina, who introduced us the concept of isomers. So I will not spend too much time here. But what is important to remember here is that the stability of the chiral axis will be determined by the substituents that you have on both sides of your axis. So if you take this example of this simple dialdehyde bi-aryl dialdehyde, the energy of interconversion around the axis here is too low to make this compound stable, let's say, in the lab. On paper, it would be chiral. In practice, it is not. What you need to do here is to add a methyl group to increase the stability of this axis. And you see, you go here from below 90 kilojoule per mole up to 124.7. That's what the calculation tells us. And indeed, if you generate this molecule and analyze it on chiral phase HPLC, you will indeed see a racemic molecule and the 2 isomers are nicely separated. So we thought, well, let's have a look if our enzymes are able to utilize this chiral compound in this disproportionation system that I just presented. Turns out that yes, they can. However, you see by the very high conversion up to 90% that at that stage we don't have any atroposelectivity. Essentially, you're going to convert your 2 isomer at a similar rate. The product appears, and that's the very interesting part. Once you cyclize those compounds, the chiral axis lose stability. So you're going down the scale on your delta G, meaning that your product, not at least with the methyl group, is not chiral anymore. So that means that we are reducing the amount of isomers that we can generate, and we demonstrate also here that we have a very favored regioselectivity for that one lactone. As we are interested in making chiral compounds here, we had to design molecules that would allow us to have lactones with a stable chiral axis. And this could be obtained by moving to a very bulky substrate that is a dinaphthyl compound. Here you see that the delta G for the product is so high that we won't have any problem here of racemization, epimerization, or just non-stability. The enzyme we screen, and I will also spare you the details here, were very active and at least very stereoselective. So we see we have kinetic resolution here with an E-value greater than 200. And one of the latest compound we synthesized here, because we wanted to increase the conversion by making the molecule smaller, is this methyl fluorinated dialdehyde. Here also the product is stable. However, it tends to racemize at room temperature. You see the delta G here computed being 113 kilojoule per mole. However, the enzymes were very happy in working on that compound. And also here we have atroposelective behavior coupled to regioselectivity so that we generate only 1 isomer. And the final data I want to share with you here is to demonstrate the racemization of that compound. So we looked at it at various temperature and could also experimentally verify the barrier here of racemization at around 105 kilojoule per mole, meaning in essence that most likely the enzyme is very selective, but we are losing enantiopurity of the product as we are working up the reaction. So that's all what I wanted to share with you today. So I'm grateful for a very dynamic and enthusiastic group of students and postdoc. You see here the list of my students and postdoc and colleagues who have contributed to these two topics. We have also excellent collaboration across Europe, especially with Italy, I would say. And I would like to finish by a short product placement. I'm associate editor at the journal Catalysis, Science and Technology, so I would really be happy to see more contributions from the field of organic chemistry, so do not hesitate to contribute. And the last product placement, I would say, is very personal. I'm looking for a postdoc, so if anybody in the audience is interested in entering this fascinating area, please reach out. I would be happy to get in touch with you about this project. So with that, again, thank you very much for your attention. I'd be happy to answer any question.
Super, Melanie, excellent presentation. And only somebody coming from France would describe something as a vignette. Very nice, great to hear that. So really super work on repurposing enzymes for non-natural reactions. Mario, you've got a question?
Yeah, thanks a lot, Melanie. Maybe I just understood it wrong. In the last example that you showed, did you say that the product is epimerizing under the reaction conditions, or the stable one, the final product, is epimerizing?
So this one indeed is at room temperature, is slowly racemizing. So you are losing enantiomeric purity over time. It's not that dramatic. You see that to really increase that, and you see the half-life as we increase the temperature becomes more problematic. So by the time we work up the system, you see the enantiomeric excess of the product is at 87%.
Mm-hmm.
Yes.
Great, thanks, Mario. There's a question from the attendees. So Robert Redmond, I'm just after clicking the button, which should allow you to ask your own question. Robert? That doesn't seem to be working. So maybe, Robert, if you don't mind putting your question in the Q&A while we listen to Rolf's question.
So congratulations for getting this atope selective chemistry to work, or biocatalysis. I have a question to your first part. So in the isomerization, you get this homoenolate intermediate. Did you think about using it as an substrate for adding an electrophile, for example, an aldehyde or an acyl donor, that you form carbon-carbon bonds instead of just adding then another proton?
Right. So exactly. So this is, let's say, for now, paper chemistry. We have not tested that. And you know very well that upon preventing protonation, we can enter a range of fascinating chemistry. So very good question. We have not tasted that yet. I do see the challenge in intermolecular reactions. We have designed, again on paper, a couple of substrates that could enter intramolecular reaction, different from cyclopropanation that you know very well. And we'll have to just, get down to try it.
Mm-hmm. Yeah, good luck.
Thank you.
Great. Any other further questions? I loved your example of beginner's good luck. I mean, you don't often get that type of, as you say, the stereocomplementarity where with one enzyme you're getting an enantiomerically pure material.
Yes, especially after having testing 20 more enzymes later on, I could tell you that was really just pure luck. So
yeah. Well, well, sometimes we all need good luck. That is for sure.
Mm-hmm.
I don't see any further questions coming through, so we'll leave it there. Melanie, thanks again. And so again, I'll pass over to Mikael. Is that right?
Yes. So we do not let Mario introduce himself. So we're now gonna hear Mario Vasa from the University of Linz. He did his PhD thesis actually there at the Uni Linz under the supervision of Heinz Falk, and was then postdoc with Alois Fürstner at the Max Planck Institute für Kohleforschung in Mülheim. And interestingly, he stopped a little bit his academic career and was a research and development chemist at DSM before he started in 2009 as an independent group leader again at the University of Linz, where after completing his habilitation, he became associate professor in 2014 and was promoted to full professor in 2020. And also handed over the burden to be head of the Institute of Organic Chemistry in 2021. And I don't want to take more of your screen time. Mario, please enlighten us.
Yeah, thanks a lot, Michi. So can you see my screen? Can you see my mouse? Perfect, thanks. So yeah, I want to go back to where we started this seminar today. I want to go back to organocatalysis now. I want to bring it back to this field. And actually, my group has been interested in asymmetric organocatalysis right from the beginning when I started my independent career. And over the last few years, we became especially interested in isochalcogenourea, so this general class of nucleophilic organocatalysts. And I want to show you a bit today what we can actually do with these catalysts. Now, I'm not sure how familiar you are with this class of catalysts. So in principle, these isochalcogenoureas are defined by this general bicyclic amidine type structure, where you have here a chalcogen atom which really strongly influences the reactivity of this nucleophilic nitrogen. And traditionally, the most commonly used derivatives in this field are the isocyanurias, so where you have a sulfur here. And those compounds have been around for around 2 decades now and been very successfully used in catalysis. And more recently, Andy Smith from St. Andrews, Scotland, also introduced the analogous selenium and oxygen derivatives. Now, what can these catalysts do? And in principle, there are two very, very well established activation modes. The first one is that these catalysts can react with different acyl species like acyl halides or anhydrides. And so you have nucleophilic attack, especially for the ITUs, there it is really most established. So the ITU bites in and you form such a chiral acyl transfer reagent. And what you get here is actually nothing else than an acyl transfer reagent, which you would get when DMAP reacts with an acyl halide. But in this case, you have here the isocyanuric unit, and this is a chiral acyl transfer reagent. And noteworthy, the whole geometry of this transfer reagent is very much defined and stabilized by a pretty strong chalcogen bond here. And because of the chirality now, of course, if you react this with a subsequent nucleophile, like a racemic alcohol or some substrate which can be used for desymmetrization, you can really very strongly control the enantiomer selectivity here, and you can use this very fine for example, kinetic resolutions of alcohols or amines, or also desymmetrizations thereof. What you can also do with this catalyst is that you can use starting materials, carboxylic acid derivatives, which are enolizable. And in this case, of course, again, the catalyst bites in, but under basic conditions, you deprotonate here and you form a so-called C1 ammonium enolate. Again, this enolate is very strongly controlled by chalcogen bonding hereby. And what you can now do is you can do classical enolate chemistry, so you can do alpha modification, where the catalyst controls the alpha functionalization with the electrophile. And then in the follow-up step, a nucleophilic species can go bite in and kick out the catalyst. And so you have then a very, very, I would say, versatile approach for doing alpha functionalizations, but also maybe cycle additions. And we have been working in this field for a few years now, and I want to show you some applications with respect to C1 ammonium enolates and what you can do. Some years ago, we became interested in doing alpha chlorination chemistry. And what we found is that when we use electron deficient aryl acetic acid esters, so we really take an ester where we have here a pentafluorophenyl ester, And we reacted with NCS in the presence of a chiral isosiurea catalyst. Then we get the alpha chlorinated products with very high levels of enantiomer selectivities. And what is actually going on in this whole process is the following. The catalyst first bites in and you form this C1 ammonium enolate I've shown you before. This then reacts with the NCS in the stereo-defining step, so you carry out the alpha chlorination. And then the catalyst, which was bound here, can be replaced by two ways. Either you directly add an external nucleophile like an alcohol or an amine to give you esters or amides, or the alcohol which was cleaved off in the first step, so this electron deficient aryl oxide, can bite back. You get this highly activated species, and this species can then be further functionalized as well. And this was actually something which we were very happy about, but there was a bit of a limitation here. And the limitation was that the reaction was actually relatively slow. And furthermore, we had to do the whole process, including the quench with the nucleophile, at -60 degrees. And the reason for this limitation is that this intermediate here is very prone to epimerization. So as soon as you warm this whole mixture up, or as soon as you do this quench at room temperature, you will see racemization. And for a while, we thought what we can do if we can maybe improve this somehow. And one solution that my student David came up with was to change the starting materials. And what he decided to do is he decided to start from alpha diazoketones. So these alpha diazoketones under photochemical conditions can undergo a very, very well-established Wolff rearrangement. So you expel dinitrogen and you form them this ketene derivative. And this ketene then reacts with the ITU. The ITU bites in. You form again the same type of C1 ammonium enolate, which can undergo chlorination, and an external nucleophile can then bite in, and you can get the same type of products that you can get here with pretty high selectivities. And operationally, these are very simple conditions. So everything we have to do is really to mix the starting materials. So the alpha diazoketone, the alcohol, and the NCS together with the catalyst. In toluene, turn on the light, and after 2 hours, everything is done. And this is something that we really like a lot, but I also have to admit that this is a reaction which is really, in this specific case here, we have been pretty lucky because these were really the optimum conditions of reactivity, electrophilicity of this reagent and reactivity of this diazoketone. What do I mean with this? If we use other electrophiles which are more reactive, then already this ketene here is directly alpha functionalized without the interaction of the catalyst, and we get racemic product only. If on the other hand, we use electrophiles which are less electrophilic, then this species here is trapped by the alcohol, and the only thing you get is the ester of the aryl acetic acid, but you don't have any alpha functionalization. So we really have, in this case, the right balance of reactivity of the reagents. Now, all this chemistry here gives you products where we have here an aromatic group. And a while ago, we thought that we can maybe go beyond and maybe also expand this chemistry to amino acid chemistry. And our group has for years been working on such glycine shift bases, and we wanted to use them to get more advanced amino acids. And the most common approach how you usually use such glycine shift bases is that you use them under non-covalent organocatalysis. So classical phase transfer catalysis, Bernstein base or Lewis acid catalysis, where you use usually bulky alkyl esters, like especially T-butyl esters, and under no covalent organocatalysis, you carry out alpha functionalization, and then you can carry out further manipulations and get them maybe into peptides or whatever. And this is something which is really powerful. And a lot of people, especially Keiichi Maruoka or Martin O'Donnell, pioneered this chemistry and really introduced a lot of powerful concepts. However, there's one thing that we don't like so much here, especially when you want to use these amino acids and you want to do a later incorporation maybe into peptides, then you always have to deal with this ester. And usually you need a T-butyl ester, which you have to cleave off under acidic conditions, and then you need classical peptide coupling strategies. And what we decided to do here is we decided to explore if it may be possible for us to use, again, a glycine shift base where we have here a more activated ester, an electron-deficient ester, use a chiral Lewis base, generate again a C1 aminoaminoate, do the alpha functionalization, and then replace this Lewis base either again with the group we kicked out here, or directly with an external nucleophile. And we thought that this should give us complementary reactivity, and this should maybe allow us to access products more easily that are not so easily accessible by the existing way. And funnily, when we started working on this, we really realized that we are not the only one interested in this. And Andy Smith from St. Andrews, who is really the front runner in chiral isocyanuric catalysis, was also looking on the same kind of starting materials. So we teamed up, and what our groups managed to do is they managed to introduce a glycine shift base where we have here a paranitrophenyl ester. It was a bit of work to make it, but now we can do this on gram scale. And the first reaction that we tested on our side was the addition of these compounds to paraquinone metides. So paraquinone metides themselves are very, very useful 1,6 acceptors, driven by the aromaticity that you gain when a nucleophile adds to this position. And our idea was that, again, this compound is activated by the catalyst. You form the C1 aminoaminoate. This undergoes a 1,6 addition where you set this bond here. You form 2 stereocenters, and then the catalyst can be released again by the group that you kicked out here. And luckily, this really worked very, very fine. So Lotte identified tetramycin, which was actually an isocyanuria that was used in vet medicine in the past. And this catalyst works very fine for quite a few different compounds. I don't want to bring it too much into the scope, just to outline a few examples, different aromatic groups here, different modification here, different heteroaromatics here, a bit less tolerated. But overall, we think it's a fairly general concept here. The Scottish side investigated other Michael acceptors to use such very, very active Michael acceptors like those, worked also pretty fine. And in their case, they immediately trapped it with an external nucleophile to give you, for example, esters here then, or amides or esters. And what they found in their case is, and this was the first example along this line here, they found that actually the selenium catalyst is more active than the sulfur catalyst. And in this case, the selenium catalyst was more reactive, but also more robust with terms of enantiomer selectivities. And I will show you another example where this is very similar in a bit. Now, I told you right from the beginning, our motivation here was to do further manipulations. And here you already see you can easily make amides or esters. And also we, for our quinometide chemistry, saw that we can use this paranitrophenyl ester and carry out, for example, trans-esterifications to give you classical alkyl esters. We can modify this and make thioesters. We can make amide bond formations. And we can also make such simple dye peptides here. So you see, actually, this can be very, very generally be applied. So this was the first part of my lecture, and I hope I could show you in this first part that these isocyanurias or these isochalcogenuurias in principle can be very, very powerful catalysts to activate carboxylic acid and do further manipulations. And we are still working very heavily in this field, but this is something where we follow, I would say, an established activation mode. And considering really the potential of these catalysts, we were also wondering very recently if we can maybe use these catalysts and expand the application scope, if we can maybe use them and activate and control other classes of starting materials, which would maybe give us some new catalysis concepts. And one class of compounds, one class of electrophilic compounds that we have been playing around in the past are alinoates. So in principle, alinoates are defined by this structure here. So you have this cumulin unit here and an electron withdrawing group here, which makes this beta position here pretty electrophilic. And what you can do, and this is well established has been well established by numerous groups, you can use them and react them with a Lewis basic or a nucleophilic organocatalyst the catalyst adds to this beta position, and you get such a zwitterionic species here. Of course, this species can also be described by a second resonance contribution. If you look at these two resonance contributions, you see that in this case, if this one would react further, it would be an alpha nucleophilic species, whereas this one would be a gamma nucleophilic species. And furthermore, sometimes you have alenoids where you have here another electron withdrawing group. And if this is the case, you can also imagine maybe that a proton transfer is happening, and that you then have these beta prime nucleophilic positions. So you see you can access actually quite a broad diversity of reactive intermediates here. And if you then give these compounds a chance to react with a dipolar reaction partner, you can really carry out quite a lot of different formal cycloadditional reactions with sometimes really high selectivities. And most commonly, this chemistry is catalyzed by either using tertiary amines or tertiary phosphines as chiral Lewis-based catalysts. And just to show you a bit on the phosphines, so these are some old examples from our lab. We have used these alenoids here, where we have here another acetyl type group, and we reacted them with different acceptor motifs. And for example, when we reacted them with these Michael acceptors, we get a classical 4+2 cycloadditions. And in this case, it's this carbon here, which is nucleophilic, adds to this carbon, and then this position here cyclizes back into this one. So it's a classical 4+2 cycloaddition, and we could catalyze this in a racemic manner with tributylphosphine. If we take the same starting materials and we react it either with these compounds, which under basic conditions give orthoquinone or with these paraquinone, we in both cases got a form of 4+1 cycloaddition. So what happens in this case is that actually this carbon here becomes nucleophilic, bites into this position, and the oxygen cyclizes back via this position. In this case, we needed a stoichiometric amount of triphenylphosphine. In this lower case here, where we had the same mechanism, we managed to do it enantioselectively in a catalytic fashion, but only with this pretty advanced and expensive catalyst. And these results were on the one hand side very encouraging for us, because it really showed us that you can do a lot of very, very nice and complexity generating chemistry with simple starting materials. But it also showed us that this chemistry is sometimes pretty hard to predict, because I really have to say this chemistry came out of pure luck. We didn't expect this product. And one more point, it showed us that sometimes the existing catalysts come to limits. We were not able to carry out these two reactions enantioselectively. And also in this case, this was the only catalyst that really, the only chiral catalyst that was really active and that gave us some selectivity. So we thought that maybe we can go beyond, and maybe we can use the isocyanurias or the isochalcogenourias in general to catalyze these reactions. And the benefit of one of the benefits would be that we also found all these isocyanurias and isochalcogenourias being very, very bench stable. So you don't have the problems that you sometimes have with phosphines, that they easily get oxidized, that they easily react with remaining dioxygen that you have under your reaction conditions. So we started looking at the potential of using these catalysts to activate alenoids. And at the time when we started this project around 3 years ago, there was really nothing reported along this line. And our first test reaction in this field was to react these very, very simple alenoids with these reactive Michael acceptors. And the reason why we used this combination was, on the one hand side, these are Michael acceptors where we know that they're really reactive. So we could be sure that they should somehow react. So the electrophilicity of them should not be a problem. In addition, we also knew that there have been already reports using this reagent combination with either DAPCO or triphenylphosphine as activators. So for example, there was a report in 2018 where they used a stoichiometric amount of DAPCO, and in a stoichiometric amount of DAPCO, they got the 4+2 cycloaddition product. Of course, they only got the racemic product with an E-configurated double bond here. If you use the same reagent combination with triphenylphosphine, you get a 3+2 cycloaddition, And again, this was only, of course, a racemic approach. So we thought it may be a nice playground. And so we started working on this. And we have to say that quite in the beginning, the results were really not successful. What we always observed is we observed consumption of the starting materials, but the whole reactions were really messy. But at a certain point, we had a kind of aha-erlebnis. So really, we had something where we realized something is going on, because what we found is that when we analyze the whole reaction mixture by MS, we find the addition product. And this meant for us that maybe we should raise the temperature, because somehow the addition may happen at higher temperature in the ESI ionization source. And as soon as we increased the temperature to 80 C, we found the formation of a very distinct product. It was again the 4+2 cycle addition product, like others got with Dabco, but in this case, we had the set configuration of the double bond. And even more interesting for us, we could do it in a catalytic manner. And this was exactly the one result that we needed. So we immediately started to screen the chiral catalysts. And first, we used different isocyanurias. So we did all the screening with 5 mol% of the catalyst at 80 C. And interestingly, we found that among the commercial derivatives, BTM, so where you have here this 5 ring, did not work, whereas the compounds where we have here the 6 ring, which are usually a bit more nucleophilic, they worked very well for this reaction, good yields, and high non-cell activities. And when we then changed for the other analogs, when we changed for the selenium derivative, the yield and the selectivity was even higher, whereas for the oxygen analog, the yield was lower and also the selectivity was lower. And with this outcome in hand, we then, of course, tested the scope. This is a bit of an overloaded scheme, so I really don't want to lose too much time here. The only thing I want to mention is that we did the whole scope with different starting materials and esters more or less twice. We did it once with the sulfur derivative, with the hyper-BTM, and once with the selenium derivative. And in all cases, the selenium catalyst was more active, gave higher yields and higher cell activities. So if you now wonder what you can do with these compounds, you can do quite a bit. If you have a t-butyl ester, you can, of course, hydrolyze it. You can carry out carbonyl reductions. So if you just treat it with lithium borohydride, you only reduce this carbonyl group. If you treat it with lithium aluminum hydride, you reduce the keton as well as the ester. And of course, you can also carry out carbon-carbon double bond hydrogenations under Wilkinson conditions. So you can modify these compounds very, very easily. Now, what is going on mechanistically? And of course, right from the beginning, we had a certain idea. Mechanism is not too complicated, as you will see in a minute. But we teamed up with Raphael Robiet, with whom we worked in the past, because there were really a few things about the whole results that were a bit puzzling for us. Right from the beginning, we have seen that we really need high temperature for these reactions. So why? And I mean, doing organocatalysis at 80 degrees centigrade is not the most common approach. The other thing is that we saw a really striking difference of different catalysts. I told you the BTM, where you had only the 5 ring here, did not show us any product, where those with the 6 ring gave us some product. So we were wondering if we could get more information on the mechanism. And what we think now what's going on is the following. The catalyst, as I proposed, adds to the beta position. You get this kind of ammonium enolate now. And we also have detected this species by MS and also in the NMR. This compound then undergoes the 1,4 addition to the Michael acceptor, gives you this intermediate. Then you have a ring closure. And finally, after a ring closure, you have the elimination, where you kick out the catalyst and where the catalytic cycle is closed. And if you now look at the whole catalytic cycle, and here is the cycle for a chiral catalyst, but we also did the same cycle for the chiral derivatives. What you see is that actually the addition of the catalyst to the alinoate is the rate limiting step. And the second step, which is very high in energy, is going from intermediate B via this ring closure to the elimination. So there are 2 energetically pretty high steps. And we think that it's these because of these two steps here, which are energetically pretty high, that we first of all need the high temperature for the whole reaction. Second, I told you about the different reactivity of the different catalysts. And we see that the addition of the catalyst to the alinoate is really very high in energy. And what we now think is what is relevant here is the more nucleophilic the catalyst, the more readily this whole reaction works. And we are now currently working on incorporating all the catalysts to the Maya nucleophilicity scale. And we see that indeed the selenium catalyst is more nucleophilic than the sulfur, and the selenium is working better for this chemistry. So we think we have a pretty good understanding for this whole process now. In addition, these calculations by Raphael also showed, also confirmed the addition, the complementary pathways for DAPCO and triphenylphosphine. He could also confirm the double bond configuration and the absolute configuration. So we think the calculations are pretty solid. Now, is this a general process? And we tried quite a few other acceptors in the past, and work is still ongoing. Just a few examples from something which came out recently. If you use barbiturates as the acceptors, you get again the same type of 4+2 cycle addition here. You have again the Z-configurated double bond here. And very interesting, if you have here an acetyl group, this is really only a spectator unit. So there's no interaction of this group in this case. If we use such CF3 enones, we get the products again in a 4+2 manner. In this case, really excellent enhancer selectivities. And we have other examples now where this works as well. So for us, it looks that this kind of alinoate activation with these isochalcogenic really is a very, very general process. Good. With this, I'm at the end. I hope I could show you a bit what we can do with these catalysts. But I don't want to close without thanking those people who really did the work. So you have seen the name on the slides already. Most people in the group are currently working on this project, but we have other things going on as well. I also want to thank the funding agency. Most of our chemistry was really very generously supported by the FWF over the last few years. And especially for this project, I want to highlight the collaboration with Raphael Robiet and Andy Smith. And with this, I'm at the end. I think I'm fine in time. So thanks a lot for listening.
I would say super, Mario, but that would be totally expected at this stage. So well done. Excellent presentation. And again, it's open for questions from panelists and attendees. So two participants have raised their hands. So Rolf, you're straight in there again. Off you go.
Thank you very much. So congratulations, Mario, for the angewandte paper in this, I think, a very potential chemistry. I have a question regarding your first part, the chlorination. Did you consider using, for example, acyl azides as nucleophilic donors? Because you would release an azide which could replace your chloride and probably getting a more stable compound. And alpha acidocarboxylic acid can then convert it to amino acid. So it's work of Dave Evans, which would work like this.
I mean, what we tried is we tried to do the direct acidation, which did not work with electrophilic azide sources, but we can take these compounds and carry out stereospecific SN2 reactions. And we did this with azides. So we have a few examples in this later paper here. So this works very nicely with very high levels of stereospecificity.
Yeah, but did you start instead of using the pentafluorophenol acid just with an acyl azide already from the very beginning?
Okay. Sorry.
Mm-hmm.
No, this is something we did not try. Sorry, I understood it wrong. Something we did not try. It's a good idea. Yeah?
Great. Thanks, Rolf. Coming through from the attendees list, we have Ravi 0020. I've allowed you to ask the question. I don't know if it's going to work this time either, but we'll have a go at asking.
Hello.
Ravi, there you go.
Yeah. My question was how strong that halogen means selenium bonding, chalcogen bonding was in comparison with the halogen bond.
Yeah. So this is something that was not done by us, but it was done by Andy Smith. So with the selenium, the chalcogen bond is a bit stronger. So if you go to this angewandte paper by Andy Smith from 2020. They really analyzed the species, and you see that with the selenium, the bond is a bit short and it's a bit of a stronger chalcogen bonding. If you go for the oxygen derivative, there is no such interaction anymore. Then it's floppy.
Yeah, but hello.
Yeah.
Yeah. If you compare with halogen bonding, which one is the strongest? Chalcogen or halogen bond?
I think it depends on the substitution pattern. It really depends what you have close to the halogen or close to the chalcogen. I'm not an expert in halogen bonding. In principle, I think the halogen bonding can be stronger, but maybe Davide is in the audience. Davide, maybe you are more qualified than me to answer this. But I would say it's very in halogen bonding, it's much more depending on, I think, the decoration that you have around. But
yeah.
Okay, thanks, Ravi. We'll leave it at that, and just we'll move over to David, who's got another question.
Okay.
David, thanks.
Hi, Mario. Thank you for the very nice presentation. I had a very similar question to as Ravi about the chalcogen bonding. Have you did not really modify, right, the different catalyst with, for example, different electron withdrawing group? Because usually when you put an electron withdrawing group, you make the chalcogen more acidic, and so then stabilize. And probably you have more effect also on the stereoselectivity.
It's ongoing.
Okay.
There are effects. We are currently we have teamed up with Andy Smith, so we have quite a bit. But maybe, David, how would you see it in principle, chalcogen versus halogen bonding? I think
it really depends on the strength of the sigma hole, on the Lewis acidity of the chalcogen or the halogen. It depends on the number of substituent, but of course they can be both of them. They can be very strong or very weak, but none of them really it is higher than the other, unless you add really a lot of electron withdrawing groups. So it depends on the substitution on the aromatic rings that bears the chalcogen. But it can be, it depends. Yeah. Thank you.
Super. Thanks, David. So again, I think we're a little bit behind schedule, so we might kick on if that's all right. Thanks again, Mario. Super. Very good.
All right. So it's my pleasure to introduce our next speaker. We are now moving to Innsbruck, and I'd like to introduce to you Professor Fabian Dielmann. He studied chemistry at the University of Regensburg in the group of Professor Manfred Scheer, and also did during his PhD studies parts of his thesis with Malcolm Chisholm at the Ohio State University and Professor Régis Réon at the University of Rennes. He joined afterwards as a postdoctoral fellow, Professor Guy Bertrand at the University of California and the University of California, San Diego as a Fyodor Leinen postdoctoral fellow. In 2013, he started his independent career at the Westfälische Wilhelms Universität in Münster as a group leader. And there he was supported by an Alexander von Humboldt fellowship, a Liebig fellowship, and by an Emmy Noether program of the EFG. Afterwards, he was visiting professor at the Université Paul Sabatier Toulouse III, and he was then appointed university professor at the Leopold Franzen University in Innsbruck 4 years ago. We're looking forward to your talk. The floor is yours.
Yeah, thank you very much. Can you hear me?
Yes.
Okay, very good. And can you also see my slides?
Not yet.
Oh, okay. Then I have to change something. Okay. Ooh.
Mario, have you stopped sharing? Because I still see your PowerPoint.
Maybe that's the reason why it's not working.
I think he left for the restroom.
Huh.
But Pat, you can take away screen sharing from him.
Yeah. So.
There's okay.
Okay, and now, yeah,
There we go.
just done.
So can you see my slides now?
Yes.
Perfect.
Okay,
Perfect.
very good. So first of all, thank you, of course, for the invitation to present something here. And you can see that I wrote a little bit red in the title of highlighting organic chemistry in Austria because we are an inorganic group. However, the boundaries between the disciplines are certainly loose. So I hope you will also enjoy what I'm going to tell you today. So I would like to change your thinking about phosphines a bit today. And Mario talked about a little bit about phosphine-mediated reaction, nucleophilic catalysis. And I would like to show you some very unusual phosphines, some very reactive phosphines. And I would like to talk about how to control the reactivity of phosphines by using external triggers as well. So we're also using them in bond activation and catalysis, and some examples are presented as well. Now this is, it's working, okay. So maybe, first of all, what are we doing in my group? We are pretty good at making reactive compounds and controlling the reactivity using strong substituent effects. And one substituents that we are using quite frequently is shown here in the middle. These are these N-heterocyclic imine groups. If you want, you can consider them as guanidine type groups. So they are strong pi donors. However, they have an advantage because the cyclic backbone allows for tuning of the reactivity. So if we have an unsaturated backbone, they become stronger pi donors. A saturated backbone would lead to less donating units. And of course, the steric properties can be controlled by these R substituents. So they're quite flexible. And why are we making reactive compounds at all, right? The simplest answer would be because we would like to discover new transformations that involve very unreactive molecules. For instance, carbon dioxide, sulfur hexafluoride, dinitrogen. So these are some dream reactions we are heading for. And another area we are interested in is the designing analogous phosphorus analogous compounds to organic compounds. Like if you look at this one, this looks like a carbonyl compound or a carbocation on olefin. And we would like to study how replacing carbon against phosphorus cations would change the reactivity of these compounds. A third area is ligand design. We try to make very strongly donating ligands and thereby influence the coordination or the properties and function of coordination compounds and catalysts. Well, today, phosphines are the topic. So luckily, I had to teach this morning and I missed the first talks, but in the last two talks, I could see already some phosphines, so I don't need to introduce that a lot. Phosphines are, of course, very important compounds, for instance, in organocatalysis, like Mario showed, but also as reagents, for instance, in Wittig reaction or Mitsunobu reactions, phosphines are used for stoichiometric transformations. They're also very important ligands, and one key advantage is that the electronic properties of these phosphines are very sensitive towards a change of the substituents. And this is also what we are using. And one example, no, if it's working. Oh, okay. There's a problem. Okay, my presentation just needs to reboot it. Let's see if that is working. Oh, okay. It doesn't look too good. Okay, I hope you can still see the slide.
Yes.
Yeah.
Okay.
You're at the intro slide, just your title slide.
Okay, yeah. I keep going. All right. So, sorry for that. okay, one reaction where phosphines have been quite successfully used is olefin metathesis catalysis. And you can see that the first well-defined catalysts, they were carrying triphenylphosphine ligands. The catalytic performance was improved by replacing these aryl phosphines with alkyl phosphines, which are stronger donors. And of course, it was not the end of the story, but N-heterocyclic carbines were shown to be superior ligands for these reactions, and especially the very basic alkyl aminocarbines. So when we started to enter this area, we had the idea to increase the donor ability of phosphines for the shown reasons, because we might discover new catalysts and new transformations. So the field looked a little bit like that. Tritose-butylphosphine was considered to be the strongest donor phosphine. And of course, carbines are stronger donor ligands. And this is now the Tollmann electronic parameter shown. So a parameter which is determined by attaching a ligand to nickel tricarbonyl, and then the CO stretching frequency is sensitive towards the donor ability. Now, when we substitute alkyl groups against these N-heterocyclic substituents, we can see that the donor ability increases significantly, not only a little bit, but we end up in an area where usually phosphines would not be located in terms of their donor ability. So with 3 of these substituents, the phosphines become extremely strong donors. And now this is just coordination to metal complexes. If one would think about organocatalysis, it would be maybe more interesting to see the basicity of these compounds. And you can see that these compounds are also extremely basic, almost as basic as the strongest neutral bases, the phosphazine bases described by Schwesinger. So we completely changed the typical behavior of phosphines. Usually phosphines are good nucleophiles, but rather weak base. So if you compare trimethylphosphine with trimethylamine, a phosphine is usually a weaker base. And if you would do a deprotonation, we would rather select an amine. But here you can see that the situation changes completely. So how would these phosphines then be prepared? There are several methods, but we usually first prepare the N-heterocyclic imine, and then it's just deprotonation with a base and the attachment to the phosphorus center. So we can make all different kinds of structure, monosubstituted phosphines, tri-substituted phosphines, and they would always bind with the phosphorus center to the metal complex, as you can see here. And also very bulky phosphines can be prepared. Some examples are shown here, for instance, where we have now mesetal substituents at these nitrogen atoms, and that lead to very bulky groups next to the metal center, similar to the diaryl system of the Buchwald type ligands. We also tested these phosphines in catalysis and showed that they are quite useful for the Suzuki-Miyaura cross-coupling reaction of non-activated aryl chlorides. And we also showed that one can use them in gold-catalyzed hydroamination reactions. And here again, you see the similarity to the Buchwald type ligands. Also, other groups are using them, for instance, here as organocatalysts in frustrated Lewis pair assisted polymerization of methyl methacrylate, for instance. And one topic what I would like to talk about today is the fact that these phosphines, of course, carry a basic site next to the phosphorus center. And we showed that protonation of this nitrogen atom has a very significant influence on the electron donor ability of the phosphines. So one can kind of switch the donor ability of the phosphine by adding an acid, which would bind here this nitrogen atom. And when I showed that to Frank Breher, he said, wow, we have phosphines that are switchable by attaching a redox active unit. And then we decided to team up and make a redox and proton responsive phosphine. And that is what I would like to present now. So we synthesized it by first making the phosphorus nitrogen bond and then attaching the ferrozine unit. And you can see that the phosphine which comes out here has a donor ability in the range of triphenylphosphine. So it's a good donor, but of course there's a mismatch. We have a strongly donating group and an electron withdrawing group that leads to the intermediate donor ability. So now if you would add a proton to that phosphine, it would go to the nitrogen atom and not to the phosphorus center. That was surprising to us. First, because usually always the phosphorus center is the most basic site, but it turns out, and one can understand it also, that if you have electron withdrawing substituents at phosphorus, then the nitrogen atom gets the most basic center. Remarkably, the Tolman electronic parameter changes quite dramatically. 24 wave numbers is the change in Tolman electronic parameter, and also the redox potentials are altered significantly upon protonation at the nitrogen center. Now, one can also oxidize the ferrocene unit by using ferrocenium as a reagent, and also that leads to quite a significant change in the Tolman electronic parameter. We also try to protonate that oxidized species, but when we do that in the presence of the ferrocene, then we get a reformation of the ferrocenium cation, and we get to this compound. So we were not able to do the oxidized and protonated phosphine, which would, of course, be a very strong high acid. So as a proof of principle, we also tested this concept in catalysis using now the catalytic hydrosilylation reaction of 1-hexyne. And without any additional reagents, only using this rhodium catalyst, one can see that there is mainly the beta E and beta Z product formed. And when we now add an acid to this reaction, we see that the reaction rate does not change a lot, but the selectivity changes significantly. While when we add an oxidant, we see that the reaction gets much faster, but again, the selectivity is changed. So we see that we have two possibilities of influencing catalytic behavior by using these proton and redox sensitive phosphines. Of course, a disadvantage is that we have to add something to a reaction mixture, and this made us think about a different possibility to switch donor ability of phosphines, for instance, by using light. So we were not the ones who invented this backbone. The first one who prepared these diethyonyl ethane-based carbines were Yam and Bielavsky. They showed that these structure are light sensitive and undergo an electrocyclic ring closure reaction, and this cyclized product is now a weaker donor. So the carbene, also here one can determine the Tolman electronic parameter, and we see that that changes by 6 wave numbers. And this cyclization reaction is reversible. Irradiation with visible light would lead to a ring opening and regeneration of the open form. So we were interested in using this redox or photoswitchable unit and attaching it at our phosphines. So this is the target compound. However, one problem was that the synthesis reported by Bielavsky was requiring 7 steps, and the overall yield was 3.3%. So that was not very promising for a PhD project. So we teamed up with a group of Frank Lorius and a very talented PhD student, Daniel Jansen-Müller, improved that synthesis significantly. So 5 steps and 32% yield is the improved synthesis. And now, since we had this imidazolium salt in gram scale, we could prepare 3 different phosphines, and the remaining steps are quite high yielding. Now, the phosphines at hand, we wanted to explore how the effect would be on the donor ability of the phosphines. And you can see that the free phosphine, the open form of the phosphine, is colorless. Sometimes the solution is a little bit purple because of some traces of cyclization product. Irradiation with UV light leads to the electrocyclic ring closure and formation of the deeply purple phosphine, which is now a weaker donor. And the irradiation with visible light would give back the open form. In the case of the phosphines, according to phosphorus NMR, this conversion is about 85% only. And you can see in the UV-Vis spectrum, depending on the concentration, it is very fast, can be within 20 seconds, but with higher concentrations, it takes a little bit long. Now, what is the influence on the electron donating ability of these phosphines? You can see we prepared the nickel tricarbonyl complexes, and it turns out that the shift of the Tolman electronic parameter is about 7.7 wave numbers. So we have a shift from here to here on the tab scale, like if you would go from a benzylated phosphine to a dimethyl carrying phosphine. Now, this change in the donor ability is significantly lower than the one I showed you before, which you would observe upon protonation. And therefore, maybe that's also the reason why we were not successful yet to find a catalytic reaction which would change, which would show significant differences upon photocyclization. So we try to find a system, but we're not successful yet. Okay, so having these imines at hand, this is just another nice catch from this project, I would say. We also wanted to study the basicity of the N-heterocyclic imines. And as you can see here, the basicity change of these nitrogen bases is quite dramatic. So the open form has a basicity, a pKbH plus value in acetonitrile of 25.7, and the closed form of 17. So the pKa value difference is 8.7. So that's quite remarkable. And you can see also here the photocyclization reaction is reversible and can be also switched over multiple cycles without significant degradation. Now, since we know from a different project that the basicity of 25.7 should be enough to capture CO2, we became interested in designing or studying the CO2 fixation with these molecules. And you can see that the open form is in fact basic enough to form this CO2 adducts. These are isolable, and the decarboxylation temperature is in the range of 70 degrees. Now, irradiation of that open form would lead to the electrocyclic ring closure. The basicity will drop significantly, and the CO2 adduct gets unstable, and we see the spontaneous release of carbon dioxide. Again, irradiation with visible light would then leave the open form. So if you look at that system, in principle, this could be the most energy efficient way of capturing and releasing carbon dioxide. Of course, it's a very complicated molecule, and one has also to overcome other problems associated with that system. But I think it's a nice academic system. Okay, so that was the part about switching phosphines. And now, I mean, Mario already talked about triphenyl of phosphine as a nucleophile in organocatalysis. And I think whenever someone is using a phosphine, and maybe everybody does that on a regular basis, triphenylphosphine is always the ligand of choice for several reasons, because triphenylphosphine is air stable, it is cheap, it has an intermediate steric profile and steric bulk, and it is a good nucleophile. So we thought, is there a possibility not to go to the very extreme electron donating phosphines, but maybe just improving the properties of triphenylphosphine? Improving for our group always means to make it a little bit more electron rich. And we were thinking about introducing nitrogen heterocycles. Well, Zipse showed a while ago that these pyrolyl substituents have a positive effect on the methyl cation affinity, meaning that if we attach these in the 2 position and the 3 position, then we would have the highest methyl cation affinity of the phosphines, and we would also expect the highest donor ability. Now, to our surprise, there are almost no reports about phosphines which carry pyrolyl groups in the 3 position. Usually the phosphines are attached in the 2 position, like the cataxium ligands by Bella. And there was only one report here where they prepared such a phosphine, but they didn't study the electronic properties. So we decided to develop a synthesis, and this is quite simple to introduce these trimethyl pyrolyl groups. We just need a base and the right conditions, and the phosphines can be prepared on a large scale. We were, of course, interested in the tri-substituted phosphine, and that turned out to be quite challenging because there are many intermediates that are formed, and the reaction usually stops at this intermediate, which is the adduct of the very basic phosphine at the disubstituted phosphorus chloride. And this can be now, by using DBU, we can make that in a one-pot reaction and on pretty good yield. Now, when we look at the common electronic parameter, we can see that these trimethyl pyrolyl phosphines are better donors than tritert-butyl phosphine. but they have a lower steric bulk. So they might be a good addition to your personal selection of favorite phosphines, maybe. We also try to make bidentate phosphines. However, it turns out that we do not get the bidentate phosphines we cannot attach two pyrolyl groups at each phosphorus, but the pyrolyl groups will bridge the two phosphorus center, which is also quite nice because such diphosphoberylines are not easy to make usually. And this is quite a straightforward procedure to make such bidirectional phosphines. Now, we were interested in using these phosphines also in catalysis. So we prepared the Krebs catalyst, the Hoveda Krebs catalyst with these phosphine. But it turns out that the catalytic activity is not very good. So tricyclohexyl is actually a better ligand in this reaction. So this is just to close the cycle because I talked about olefin metathesis at the beginning, not a spectacular result. So donorability is not everything. Well, so now this was about phosphine ligands, and now I would like to talk a little bit about using these very basic phosphines on themselves, so without a metal center. And of course, I already showed you the basicity that these compounds can obtain. And that means that these compounds are very reactive. Of course, not only one could use it for deprotonation reactions, but there are cheaper bases around. What would be more interesting maybe is to use the intrinsic property of the phosphorus atom. And for instance, we showed that we can make carbon dioxide adducts with these phosphines. However, the phosphorus, of course, can be hypercoordinated. So the adduct with an amine would lead to a stable adduct. This is known. One can buy these DAPCO adducts, for instance, of SO2. With phosphines, no stable adducts have been reported, but we showed that these adducts can be isolated first, and then we have, of course, the intrinsic property of phosphorus that hypercoordination is possible, meaning that the oxygen atom can approach the phosphorus with a rather low energy barrier, and this leads then to the liberation of sulfur monoxide and the formation of phosphine oxides. An even weaker Lewis acid would be carbon dioxide. Here we also show that depending on the number and nature of the substituents, we can form isolable CO2 adducts. With the most donating phosphines, these can even lead to a breakage of the carbon-oxygen bond. And more recently, we showed that due to the very nucleophilic nature of these phosphines, we can also compete with carbines for chloronium ions. So what did we do here? We use these phosphines to generate N-heterocyclic carbines, and the principle is that the phosphine is attacking at the sigma hole of the carbon chloride bond. And in the end, the ligand wins, which has a higher chloronium ion affinity. And the very basic phosphines have definitely higher chloronium ion affinities than carbines, and therefore the chloride is transferred to the phosphorus center. So we can use this method in order to generate N-heterocyclic carbines starting from chloroazoleum salts, and also generate very unstable carbines like this one, which would spontaneously dimerize. And when we use a very bulky phosphine like the one I show you here on the right, this phosphine is so bulky that, as you can see here, the phosphorus is buried by these tert-butyl groups that would not bind to transition metal atoms, but it can accommodate a chloride atom, and therefore one can use it in a one-pot reaction, adding this phosphine, a chloroazoleum salt, and then directly attach the carbene to a transition metal complex. So a last topic I would shortly like to talk about, because we are getting more and more interested in that, is the activation of a very inert compound, and that is sulfur hexafluoride. Sulfur hexafluoride is a very useful compound. It is an octahedral compound, and it is, if you want, the perfect example of a chemically inert compound, because the sulfur atom is surrounded octahedrally by the fluoride atoms. Therefore, this compound is extremely inert. It's not thermodynamically stable, but it's inert. So it is used on a large scale, as you can see, mostly by the power industry, in high voltage power applications. But there's, of course, a drawback to that molecule because it's so stable and it's very efficient in absorbing IR irradiation. It is the most potent greenhouse gas, so it should not end up in the atmosphere. However, due to the chemical inertness, it does more or less, all SF6 which is produced end up in the atmosphere, and therefore it's getting more and more debated if that should be used at all in these applications, and replacement compounds are looked for. Well, two challenges are associated with that. There will be a lot of SF6 and is already on the market, which needs to be decomposed. We need more energy efficient degradation methods. That is one topic. And the other one is, it would be maybe interesting, instead of degrading it at high temperature, it would be interesting to use it as a reagent for fluorination reactions. And this is something I would like to share with you today. So we came into this area by, again, using our phosphines, and surprisingly, depending on the number of substituents, we get totally different selective fragmentation. And with one substituent only, we get a fragmentation into the phosphine sulfides and the difluorophosphoranes. But when we have 3 of our donor substituents attached to phosphorus, we observe the formation of only the fluorophosphonium cation and the SF5 minus anion. So only one fluoride cation is abstracted from sulfur hexafluoride. This, of course, looks a bit strange. How can we want to abstract a fluoride cation? And maybe a comment on these SF5 salts. So some have been prepared. They are not very well studied yet, because usually they're very unstable when they are when these salts are formed together with alkali metals. They tend to eliminate alkali metal fluorides and form SF4. So in our case, we have an advantage. We have a large organic cation, and therefore these salts are quite thermally stable. So what is the mechanism behind that reaction? You can see here, this is the computed mechanism with only one substituent. So we have an energy barrier for the fluoride transfer, and you can see that this can be considered as an SN2 type mechanism. So in the HOMO of the transition state, we see that the lone pair at phosphorus is overlapping with the sigma star of the sulfur fluoride bond. So that is what we would expect for an SN2 type reaction mechanism. And we transfer formally the fluoride cation to the phosphorus center, and the SF5 minus anion is then left behind. Now, the selectivity is decided here. If we have 3 donor substituent bound to phosphorus, that means this compound is quite stable, and the difluorophosphorane gets unstable, and therefore we end here. If there's only one, you will have a complete fragmentation and formation of SF4, and that, of course, is more reactive than SF6. So what can we do with these compounds? And of course, this one is the most interesting one, because the character of sulfur hexafluoride is still preserved to some extent. Of course, we are now at the oxidation state, of the formal oxidation state +4 at sulfur. And therefore, we wanted to use this compound in oxidative fluorinations. Now, it turned out that it is the best idea to attach, again, these very bulky substituents here at phosphorus, because the tert butyl groups would make the cation even more inert, so that we can use that reagent also at very high temperature. Some reactions that we successfully use that for instance, we can fluorinate elements like selenium, tellurium, arsenic, antimony can be fluorinated. And what we observe is that the fluorides are transferred, for instance, here to the selenium pentafluoride anions are formed, and elemental sulfur. Maybe more interesting is the fluorination of organoselenium and organotellurium compounds. For instance, the diphenyl ditelluride can be fluorinated to give this phenyl tellurium F4 minus anions, which have not been characterized before. And if one would like to carry that even further, one can oxidize now, introduce an oxygen atom, go to tellurium 6, and therefore a more inert form of tellurium. And one can even alkylate the oxygen position, for instance, with benzyl bromide. And therefore, we can generate new functional groups, which are actually stable in the presence of water and air. So they might be quite interesting. So at the same time, the group of Sepp Riedel prepared the cis derivatives on a different route, and also showed that these are quite interesting functional groups. Now, can we also use these SF5 salts in classic sulfur-based deoxyfluorination reactions? That is something we also wanted to study. But first, maybe to go back to the previous slide, the reagent, of course, we are using is quite complicated because the phosphine needs to be prepared, and then the reaction with sulfur hexafluoride would give the reagent. So we thought, is there a possibility to simplify the phosphine? And I think this is the most simple phosphorus superbase that one can prepare. And I show you the preparation here, therefore. So these phosphines can be prepared by mixing simply 3 liquids. So phosphorus trichloride is a colorless liquid. The tris dimethylaminophosphine is a colorless liquid, and the tetramethylguanidine is also a colorless liquid. All of them are commercially available. So you mix them in the right stoichiometry. A little bit of acetonitrile is needed, and then you would have the liberation of dimethylamine gas, or that would go out of the reaction mixture, and we form the protonated phosphonium chloride here. This has been prepared on a different route by Schmutzler already more than 20 years ago. But Schmutzler was never able to deprotonate, probably because he had some lithium salts impurities. Because the deprotonation, as we showed, needs to be done at 70 degrees Celsius with KHMDS, and then one can produce this phosphine in a very efficient manner and on a large scale. Now, this phosphine is a crystalline material, as you can see here. It has a very high pKbH plus value, so it's a very strong base and also a good donor ligand. But of course, what we were interested in here is the reaction with sulfur hexafluoride. It would not react spontaneously with sulfur hexafluoride, but irradiation gives the product that we were looking for. So the SF5 salt, you can see in the fluorine NMR nicely the pentate and the doublet of the SF5 anion. And here one can see that this is a crystalline material as well. Now, how does this perform in deoxyfluorination reactions? We have tried some reactions, and we tried also to compare that to more established reagents like dust and Stahlfluor. We did not use any additions here, so only the plain reagents. And of course, we know that there are improved protocols by having additional additives, but we wanted to have the direct comparison. So we only used the one equivalent of the sulfur-based fluorinating reagent and one equivalent of the substrate. And one can see that both SF5 salts are quite effective in generating acyl fluorides out of carboxylic acids or carboxylates and even amides and anhydrides. So that was quite surprising. Also, that is working. And for the deoxyfluorination of alcohols, they are also quite effective. Aldehydes and carbonyls, the reaction is quite sluggish, and we mostly get the elimination products. And this is maybe also what is expected. Else, of course, there's a lot of information here, but I don't want to go too much into detail. Bottom line is that these reagents can be used in deoxyfluorinations. They have one disadvantage when we use an acid, and you can see that here, the substrate slowly decomposes the phosphonium ion. So an acid would decompose the phosphonium ion at higher temperature, and therefore the addition of acids or acids in the reaction media are not tolerated by these reagents. However, due to the thermal stability, we can apply these reagents at higher temperature, and that is maybe a benefit. So for some applications, they might be useful. Okay, with that, I'm already at the end of my talk. This is kind of the Hall of Fame of our most basic phosphines. Each of them has its own characteristic, and is also, one has to know how to make them. But of course, if one makes them, they are quite rewarding because they're very reactive. However, using them in aqueous solution is only possible for the very right one. The other one are sensitive towards hydrolysis due to the high basicity of the phosphorus atom. This is my group. Of course, I'm very grateful for their passion and for their work. They did all the work. I'm just the supervisor. And a last comment on a possibility, if you are interested in pnictogen chemistry, a possibility to share your results of pnictogen chemistry will come up in February. So there will be the European workshop on pnictogen chemistry held in Innsbruck in this building. And you can see in February, Innsbruck usually looks like that. So it might be also quite attractive to go there. With that, I'm at the end, and thank you very much for listening, and I'm happy to answer questions.
Super. Thank you very much, Fabian. Again, open for questions. Mario, you're straight in there. Off you go.
Yeah, thanks a lot, Fabian. Fabian, I have a practical question. How do you measure, or how do you determine the pKa's?
Yes, so the pKa's were determined, I showed different ones. So the one with the phosphines were determined actually by NMR spectroscopy.
Okay.
So we used compounds that have a similar basicity and then used phosphorus NMR spectroscopy in order to determine the pKa value.
Mm-hmm.
And the one of the nitrogen superbase was calculated.
Okay.
However, we used an experimental value as a reference to be as accurate as we could get.
Okay. Okay.
Yeah.
Thanks.
Okay.
Good. Those 1,2,5 trimethylpyrolyl systems were very interesting. Can they be converted to salts to make those ligands potentially water soluble?
Yes, I mean, very good idea. And we have not looked into that, but I guess one could introduce an anionic substituent at the nitrogen before, and then we would have water soluble phosphines. Totally right. Yeah. Yeah. Good idea.
Very good. I'm worried about the time. So I don't see any questions coming through. So Fabian, might leave it there if that's okay. Excellent work. Again, congratulations.
Thank you.
And finally, back over to Mikhail.
All right. So it's my pleasure to introduce our final speaker of today. We're moving back to Vienna and Vienna University. And it's my great pleasure to introduce Davide Bonifazi. He studied chemistry at the University degli Studi di Parma, was a visiting PhD student at the Weizmann Institute, but actually got his PhD degree at ETH Zurich by working with Professor François Diederich. He stayed also there for a short postdoc and then moved to the Foundation of Research and Technology Hellas in Patras in Greece, before he did another postdoc at the University degli Studi di Trieste. Regarding his academic positions, he started as associate professor at the University degli Studi di Trieste and concomitantly actually at the University of Namur in Belgium as junior professor for organic chemistry. Before then becoming professor of organic chemistry at the very same university, and then chair professor at the University of Cardiff in organic supramolecular chemistry. And in 2020, he moved to Vienna and became there at the Institute of Organic Chemistry, the chair professor in organic chemistry. Davide, we are looking forward to your talk.
Yeah, thank you. Do you see my slide?
Yes.
Very good. So thank you for the organization and the chance here to represent the Institute of Organic Chemistry at the Univien here in Vienna. So my group is a synthetic organic chemist, but we are focusing on the preparation of functional organic molecules through targeted oriented synthesis. And our idea today, it is always to prepare molecules, as I said, that has function. And in particular, we are very much focusing on molecules that they are colored. So when you take a molecule and is colored and has a homolumo gap, which is falling in the visible range, then what you do as an organic chemist, and when you want to change the properties of the molecule, what you do, you change the homolumo gap. And so then when you change the homolumo gap, then you change all the optoelectronic properties of a molecule. How do you do as an organic chemist changing this homolumo? There are a few approaches. The first one, it is the conjugation length. What you take a molecule, you extend the structure of the molecule, in particular the pi conjugation. Or you take this pi conjugated molecule and you start and you bend it, you twist it, and so then usually you open homolomo gap. Or what you do, you add a donor and acceptor, and then you get a push-pull system. All these effects contribute to the tuning of the homolomo gap. But when you tune the homolomo gap, you tune also the properties of the excited states, because when you excite one electron from the HOMO to the LUMO, actually you bring your molecule to an excited state. And so the excited states that you are involving in the HOMO-LUMO are singlet or triplet. And so when you excite and then you go to a singlet state, then the singlet state can deactivate to a triplet, and then you can have fluorescence, you can have phosphorescence, and then this can be used in the optoelectronic device. But not only. When you are at excited states of in a molecule, then essentially you may have also bioradical behavior of these excited states that has different lifetime, different properties depending whether it is a singlet or the triplet. And then these excited states, you can use it, for example, to do chemistry. And so essentially, this bioradical behavior can be used to generate energy, can be used to generate chemical reaction. And then what is very important when you analyze this type of processes, it is that you need to measure the quantum yield of your reaction, which can be the quantum yield for the excited states or the quantum yield of your reaction. Now, how do we control this, and how can we make these light-induced processes efficient? And one of the problems in the field, it is that, for example, if you want to devise a photoorganocatalyst, for example, what you do, usually you take your molecules, you excite the molecules at the maximum of the absorption, and then you look at the reactivity that this molecule may manifest. But one of the problems is that you have always to have LEDs. So there are really little examples in which you use solar light to trigger conveniently a reaction. And this is actually what nature does. If you take nature's leaves, you see that in autumn leaves becomes very colored, because essentially most of the chlorophyll wash it away, and then you have all the other pigments which appearing, and this pigment constitute what is called the antenna. So essentially in a molecule, in a leaf, what you have an ensemble of different chromophores which are organized in such a way that they absorb light, and then all this light, it is stored in excited states, and these excited states, the energy of these excited states are transferred to the neighbor molecules until it's funneled to a reaction center where you have actually the photocatalytic event. Now, when we focus on the antenna, of course, has a few important roles, which is the absorption of the light, which has to try and to maximize the absorption of the light. So it has to take a lot of all the energies, all the light that comes on the Earth has to try to maximize the whole spectral absorption. This is because you want to use all the possible solar energy. And it funnels efficiently and so very fast to the photocatalyst, so then the photocatalyst can start again a new photocatalytic cycle. So this is what is the antenna. And it is because light comes very diluted on the Earth, and so if you want to make it exploitable, this light you want to really use also for high energetic transformation, you need to try to get all possible wavelengths, solar wavelengths. And so this is how does it look like. This is the light that hit our Earth, and this is what it reaches the leaves, and this is how the chlorophylls absorb. So you see that where the plants absorb are in this region around 400, and this is where you have the highest concentration of light. And so this is what plants try to optimize. So how do plants do? So it is a very simple system. It is a very schematic. So essentially what you have two parts. You have the reaction center, and then you have the light harvesting system. All these are, let's say, confined in a membrane, and in this thylakoid membrane, and then essentially the light harvesting is the duty, it is to absorb the light and then to funnel it to the reaction center. So when you look at this reaction, this light harvesting system, you see that they're composed of different chromophores. And those are the ones that I show you that they appear in the fall when the chlorophyll goes away. And then actually all these chromophores are there to optimize this light. So how this absorption, how this is organized? It is organized in which you see a peptide scaffold, and this protein essentially organize the chromophores in such a way that they are rigid but sufficiently close to allow efficient energy transfer, which usually are really, really fast. Some of them are in the femto and attosecond speed. And the other purpose, it is that you have different chromophores because you want to broaden the absorption of light from these 400 up to 600. And so this is how the nature works. So now what we wanted to do, we started a few years back to try to mimic what nature does in terms of constructing a light harvesting system that could absorb solar light and then could be eventually used into the transformation such as photocatalysis. And then one of the possible approaches that you can use to build such a complicated system are essentially two. One, it is a sequential route where you take the single chromophores, you attach one after the other one, and then you build up your scaffold. Or the other way, it is what nature does. Actually, before you prepare a scaffold, and in this scaffold, you just add recognition moieties, which are complementary in terms of selectivity with the other chromophores, which then are organized precisely in each different position, following the information that you use to program your template. And so here the idea, it is that you organize the different chromophores in a sequence, so then you optimize the energy transfer, and then eventually now the excitation of the photocatalyst. And so by mimicking what nature does, we initially design a system in which you have this, you have an alpha helix, and then we equip alpha helix with the modified amino acid in which we could add sulfur, hydrazone, and the diol to be used as a reactive complementary unit with chromophores that bears the complementary reactive groups. For example, boronic acid that could go reaction with the diol. You go with an aldehyde that could go with the hydrazone, formation of the hydrazone, and then a diol that you could undergo formation of the disulfide. And so we choose these three chromophores at the beginning because those that were absorbing in this region, and so to try to cover as much as we can this visible region. And then we find the condition to run these three reactions simultaneously without having crosstalk or byproduct. And so in this way, what we found that in DMF, with this amine, in 2 hours, you just simply mix the three chromophores with the template, and then you have the installment of the chromophore on the template. And then essentially you form the disulfide, the other one you go with the formation of the hydrazone, and then you get the boronic acid. And so then this is very simple way to synthesize, because essentially once you assemble your chromophores on the template, what you get here, it is an example. These are the mixture of the chromophores that you can simply remove. This is a GPC column. And then here you have your antenna that absorb the light. You see that is reddish. The color here depends on the ratio of the different chromophores, and then it can be very easily separated in one step. And then we want to study, of course, how efficient are these systems to transfer energy. To make a long story short, essentially what you do, you just excite one chromophore, and then you interrogate the chromophore that is absorbing a lower energy, and you see how much is the transfer of energy. As you can see here, only 5% of the energy that was used to excite the yellow or the red chromophores was transferred to the blue, meaning that this system was not really efficient. And so we started to investigate a series of possible structural parameters to see whether we could improve this energy transfer. And then, for example, we look at the effect of the chromophores. So we change the type of chromophores, and then we could increase one of the two transfer of energy up to 20%. We also swap the position of the different chromophores, and then again here we could improve a little bit one of the two processes, but not both. Then also we start to change the distance. So to go from 1 nanometers to 1.5 to 2 nanometers, but again, we did not really add a big improvement. And then we try also to change the solvent, and this is, we observe a certain improvement, and also we try also to modify, so in such a way that we could change the direction of the chromophores, and so then eventually to see whether it was an effect of resonance between the different chromophores. But again, that was not really impactful. But then when we look at the structures of the molecules that we prepare, and we did some simulation, what we observe, it is the following, that we got this alpha helix, and then in all the system, in all the different conformation, we observed that always these chromophores, essentially they were collapsing. And so basically what we observed, it was quenching of the excited states. And so essentially these chromophores, they were quenching each other, regardless of the distance, for example. And this is because the alpha helix template, it was so rigid, then pre-organized the chromophores to stay on the same side, and then essentially through hydrophobic interaction, those that were collapsing. And so this was a problem. And so using this template, we could not really solve this problem of quenching. In nature, again, remind you, in nature, these systems are in these proteins, but is inside of pocket, and they're really rigid, so they cannot really move to approach each other and then to quench the excited states. And so what we decided, it was to do another approach and then to take a loose architecture. So we synthesized this simple and shorter peptide, and then this peptide was functionalized with different reactive group, but this peptide, compared to the other, did not pre-organize, and so essentially it is disordered. So what we thought, instead of making something really pre-organized, we go on the opposite and we increase the entropy of the system so that all the chromophores could adopt all the possible conformation, making the system really entropically disordered. And then this was one, and then we extended also the number of chromophores up to 5 to see whether we could better maximize the absorption, all the spectral solar absorption. And so this is what we devised. So on top of the 3 bond that we used before, we added also 2, let's say in this case, non-reversible bond, which is the formation of this cycloadduct. So we went through with a kind of click chemistry with the substitution of a strain triple bond or a strain a double bond. And then again, simultaneously, we could assemble the 3 chromophores together on the same scaffold with no crosstalk reaction, with almost 100% yield for each of the 5 reactions that we ran simultaneously. Now, which one are the chromophores? And so the chromophores are those 5. So on top of the pyrene, of the PDI and of the NDI, we added also perilene and we added a cyanine. And so those, as you can see now in this slide, so those absorb all over the solar spectrum, starting from 350 to 700. And so this would let's say, assure a complete absorption of light in the spectral region. And we ran also calculation, and in this time, what we observed that there is no conformation in which you have the 5 chromophores that simultaneously collapse and gives this pi-pi stacking type of interaction, which would quench completely the excited states. But if there is always a conformation in which if 2 goes together, the other 3 are not separate, are far apart. And so this was very good for keeping the excited states of the different chromophores alive. And so here you see how does it look like at the UV visible spectrum. So basically you see that there is it is the orangey one, and so you see that it absorbs all across the UV the solar region. And then what we measure the different properties. So here also it is how does it look like. So those are the 5 chromophores. This is the mission of the 5 chromophores absorption. So this is how they look like, and it is when we have 3, when we have 4, and when we have 5. And so when you see the 5 mixture, so you see that it's palely greenish, and this is resemble the color of a leaf. And then when you see the emission, it's palely white, because essentially it tells you that all the chromophores are emitting. So all the singlet states, and partially also the triplet, but mainly the singlet states, are fluorescent because they are not quenched. And so this was really good results, because it tells us that our approach of having something that is completely disordered works as well. And then at the end, without going too much in the details, in collaboration with the group of Nicola Maroli in Bologna, so they analyze all the different process and of energy transfer with the different chromophores. And essentially what they observe, that all the chromophores talk to each other, with the pyrene being the highest energy, gives the energy to the perilene, which gives to the PDI, to the NDI, and the 2-cyanine. And the cyanine acts as a final emitter, or can transfer the energy also to a triplet of the PDI. And so essentially you have a cascade communication between the energy from the singlet, or from the highest energy pyrene, down to the cyanine, which is the lowest. But then what was important, how much is efficient it is this energy transfer. And actually we calculated that there is an efficiency that is around 92%. What does it mean? That 92% of the light that is absorbed by the antenna, it is efficiently transferred between the chromophore down to the cyanide. And it is a really important result, because it tells us that we can have an antenna that takes 90% of the light and can funnel to a chromophore to do a certain catalysis. Now, which kind of catalysis we could use this antenna? So now what we are doing, we are trying to couple this antenna to a catalyst. And what would be the catalyst of our choice? So what we are interested in our group, because we like to do this chromophore, so we are quite bidimensional, and so we are looking for a planarization method, and in particular carbon-carbon bond formation, to make these chromophores. And then when you think about making a carbon-carbon bond between 2 sp2 carbons, which could be, for example, between 2 phenyl, the number of choice of reaction are essentially limited to these 5 cases. It is the oxidation, a Scholl type reaction. So you use a strong oxidant, you form a pi radical cation, or you go with the photochemical cyclization, or you go with the metal catalyzed cyclization. So in both cases, in this case, you need, in the first case, you need a strong oxidant. In this case, usually you need a strong UV light. And when you go with the metal catalyzed, instead you need, of course, a catalyst, a metal catalyst. Another 2 possibilities are to work with highly reactive species that could be a phenyl cation or a phenyl radical. And so these highly reactive species can then undergo a carbon-carbon bond formation and then planarization. So among the different systems, the one that is less investigated has been the radical, because there are some difficulties to create these radical sp2 carbons. And so we challenge this project, and in particular, we wanted to see whether with one of these chromophores we could do this transformation. And so it is a while that we work with this chromophore, which is called PX6. PX6, it is this oxygen-doped equivalent of the anthanthrine, which is the one that has 2 carbons, and this is very cheap. This is a very old molecule discovered by Pomeroy, 1926 something, and then it's very bright yellow, very robust. And it is quite good in the formation of the singlet, so you see the quantum of the fluorescence is around 60. But most important, it is a very strong reducing molecules in its excited states, and in particular in the singlet. And so what we did, we tried to see whether we could use these molecules, that it is called PEEK6, to induce photocatalytically a formation of a radical phenyl radical species that could undergo carbon-carbon bond. And then, so we screen a different condition, and then we could observe that in DMSO, by adding around, we have a 2% mole of PEEK6, we can get 70-80%, between 75 and 80% yield in this carbon-carbon bond. One of the challenges here, it is that when you form these polycyclic aromatic hydrocarbons, some of those tend then to quench the excited state of your photocatalyst, then killing its photocatalytic activity. And so this was one of the challenges that we had to face, but then we overcome, especially with the choice of the solvent. And then, so then we had the substrate scope where we show that this reaction is quite versatile with different substituents, with different aromatic groups. And so essentially here we could do, we could change the position of the cyano. We could add 2 cyano groups, but also we could, this is compatible also with electron withdrawing group. And then also with other polycyclic aromatic hydrocarbons where we could, for example, fuse a pyrene and an anthracene, and then also to form 2 carbon bond with pyrrole and then these dimethyl benzene derivatives. How does it work at the mechanism of the reaction? The mechanism of the reaction works with electron transfer. And so essentially the PEEK6, once is excited, you give 1 electron transfer to the electron poor moiety. And so you get this pi radical anion, then through elimination of a chloride, then you form your radical benzene, which then undergo carbon-carbon bond formation, and then oxidation, which at the end you get a cation, which then eliminates in the presence of the base. One important thing is that this method works when you have an electron withdrawing group, can be a cyano, can be an ester. This is because you need to favor the electron transfer to the electron-poor benzene rings. The system does not work when you don't have the reaction, when you don't have the cyano groups or this electron withdrawing group. And so this is what we are trying to do, to develop a new photocatalyst that could also trigger the reaction with unsubstituted systems. Yes, and then what we are trying to do as well now is to couple our photocatalyst with the antenna, and so to see whether we are able to run the reaction under the balcony of our institute. So, and then at the last point, at the last section of what I want to tell you today, it is instead to see whether we can use this chromophore for another application which goes a little bit more in material science, and to see whether we could exploit these chromophoric properties of molecules for information encoding, and in particular data storage. So now let's imagine that we have a system that we can react 2 molecules, and this is when you react the 2 molecules, you make a reaction, and then you quench, for example, the excited states of a given molecule. So we call it as the forward reaction, and then you block the fluorescence, and so the molecules, the adduct, it is non-fluorescent. But then this reaction should be reversible in such a way that then you, again, disassemble the adduct in its original component, and then you get, again, the fluorescence of the molecule. So if you are able to make this n times, essentially you can attribute one information to one state, that it could be the reagent, if you want, and then to another state, which are the product. And so if now you imagine that you take this and then you put on a surface, and then you are able to do this reaction locally in the different position, then essentially you can create a matrix to which you can assign, depending if it is fluorescent or no fluorescent, a state, and then actually you can write in a binary code, and then you can store information by triggering an organic reaction. So what we thought to do, it is to use one quite old reaction, that is the formation of an endoperoxide. Usually endoperoxide are formed between, for example, anthracene type derivatives in which you add a photosensitizer, and this photosensitizer produces singlet oxygen, then theoretically react with the anthracene to form the endoperoxide. Now, if you use a sensitizer, then it means that you have to use at least 2 molecules because your molecule plus a sensitizer. Our idea, it was different than instead to use an anthracene derivative, which then can also self-sensitize the formation of the singlet oxygen. And then once you sensitize the formation of the singlet oxygen reacts with the molecule itself to give you the endoperoxide. Since you form the endoperoxide, you break the conjugation. And usually when you break a conjugation of these pi conjugated molecules, what you do, you have two effects. What I told you at the beginning, you increase the homolumo gap. And so basically, you make the molecule often uncolored. And the other things, you break the fluorescence. And so this was our idea. It is to devise a molecule that we can excite, produce singlet oxygen, and this singlet oxygen react with the very same molecule to give you an endoperoxide. But then the endoperoxide can be broken with temperature, and so you release again singlet oxygen and your anthracene. So with our experience with this molecule doped with oxygen, we know that when you add a Pirano ring to an organic molecules, to a pi conjugated molecules, you introduce some vibrational mode, which then favor intersystem crossing, for which from a singlet, you populate also the triplet. And so this is typical. So whenever somebody wants to use an organic molecule that has a lot of triplet, you should always add a Pirano ring. And so this is usually trigger the population of the triplet. And so we come up with the idea to prepare this molecule, which has these 2 Pirano rings, and then you have an anthracene skeleton in the middle. And so this is the synthesis. This is pretty straightforward. Also, this was important for the application. So you prepare the Grignard of these methoxyphenyl derivatives. You react with the dichloroanthraquinone. You get these dihydroxylated derivatives. You do an elimination reduction with tin chloride, and then you get the full aromatization. Then you deprotect. Then you go with a nucleophilic aromatic substitution by heating. You form the 2 oxygen rings, and then at this point, when you have oxygen, you irradiate. So essentially, your starting molecule that it was colored, and this is also, we have the crystal structure to show, the tension of the ring. And then that provides as well high reactivity toward the singlet oxygen. And then when you expose to the singlet oxygen, you see the molecule become completely transparent. So you broke the conjugation. And then here we got also the crystal structure where it shows the formation of the endoperoxide. So these both molecules are stable, and even also the endoperoxide. The endoperoxide, only when you heat at high temperature, then essentially you go with the cycloreversion, and then you reform the starting material, which is then again colored. So for our application, if we want to write, if we want to cancel, we need to know how fast is this process of this type of reaction. And so then we study the kinetics through UV visible and emission spectroscopy. And then what we figured out, that actually the formation of the endoperoxide through the self-sensitization approach that we use for our molecules, it is exactly the same as when you add methylene blue as a singlet oxygen sensitization. So you see in this first part of the curve, of the graph, the speed of the reaction is exactly the same. Only that then at a certain point, when you don't have the self-sensitization, external self-sensitizer, the reaction gets simply slower because, of course, you reduce the amount of your starting material, and then there is less singlet oxygen. Then, much slower, it is the cycloreversion. So you see here the kinetics, we are around in the minute. And this is quite typical for this cycloreversion reaction. Then we also study the kinetics as a function of this power of the LEDs, and then also we measure the fatigue. So how many cycles we can do this oxidation and then cycloreversion reaction. And as you can see here, we did it more than 10 cycles, and this is, it was quite well. And then what we are going to do then we wanted to see whether this was working. And so the idea here it is that we take the molecule, we put in a polymer, as we did, and then we throw it on a glass. Then we took a mask. So you put a mask, then you take a lamp, and then you see whether the reaction undergo under solid state inside of the polymer. And as you can see here, you do, where there is the mask that leaves the light going through, it becomes transparent, all the rest remains cold. And then if you heat, then you cancel, and then you go back to your original material. Then also we have to characterize, to make also some lifetime measurement to make sure that all these are the structures. And then we could even print on a flexible substrate. And so which tell us that we could even write now on plastic, on a bottle, on a plastic, on any type of system that could be flexible. But okay, this is macroscopic. This is a little game. Can we do something that is more, it goes closer to what it could be a real map? And so in collaboration here with Dr. Trupke at the Austrian Academy of Science in the quantum physics group, so they have a system in which they can use a confocal to direct laser writing on a surface. And then what can they do? They can excite dots very precisely on a surface, and then they can make these dots. But once they make the dots, also they could probe the emission. And so basically what they do, they could excite in different regions where we wrote, and then read whether it is emitting. And so where it is exciting, where there is when there is the endoperoxide, there is no emission. When it excites where there is the molecule that has not been reacted, there is emission. And actually, with this technique, what they could do, it is really to draw dots. The size of the dot is around between 1 micron, 2 micron. And so now we are trying to go much smaller. And then you see to make 4, to make 6, or go 4 times 4 matrices, or 5 times 5. But then the most important to see whether we could also eventually cancel. And so if you take this surface here, we just did a scratch, just only to see if we were able to find where we wrote. And then you write. So we wrote this micrometer hexagon, again with the laser. And then you erase 70 degrees, 1 minute, and then you go. But then can we do really data storage? And so this is what they were able to do. This is what they wrote. So it is, again, tell you this is where you have the endoperoxide. There is no endoperoxide. And so then if you take this writing, you put in a matrix, you can say this is 1, 1, this is 0, this is 1. So this is the matrix that you obtain. And then when you convert it in the logo of the Austrian Academy of Science, this is the logo. How much is the storage space? We are around 16 gigabit, which means some of you are younger but never worked with the floppy disk. So this is around 740 kilobit. With the CD, we are around 0.90. Then with the DVD, 2.2, and then the USB that we are using, we are around 4.5. So you see that with our very simple system, we are already in the range of a CD-ROM as a type of storage. So now we are trying to improve and to get also different molecules to do this job even better. And so I hope that with these 3 examples, I gave you a glimpse of what we are doing and how we can use organic chemistry to exploit colors to do some functionalities. At the end, I thank all the funding that gave us the possibility to do this research and my research group, and thank you for the attention.
Super, David. That was an excellent presentation, and certainly the most colorful presentation that we've had today. So well done on that. Again, open for questions, and just 1 or 2, if that's okay. We're quite a bit behind time. As it turns out, I have another meeting starting at 1:30 Irish time, that is. So any questions from panelists and or attendees? Seems you've knocked them all back, David. In terms of maybe the last thing you talk about, the laser photo writing and data storage, you know, what do you need to get to before it's really a quantum jump in terms of data storage?
Yeah. So what we need to go to 20, the physics that we need to go to write to 15 nanometers each dot.
Right.
And then we will get the same storage capacity of the actual magnetic memories, the USB that everybody uses. Okay. The difference that we can print, we can write on any substrate. Yeah.
Right. Okay. No, very good. Okay. So perhaps if I'll just stop sharing there and we'll just wrap up for today. So this has been a really excellent further edition of the Highlighting Organic Chemistry series across Europe. And certainly as an external person looking in on chemistry in Austria, I always had the feeling that it was very strong. And this has certainly been reaffirmed by the quality of the talks today, but also the breadth of chemistry. And, you know, many things coming through, you know, at the start of it, Michael gave a very good overview of the strength of organic chemistry in Austria. And I'm sure he and colleagues in the society had a hard job to whittle it down to just you 7 people presenting today. So that's an unenviable task, I would say, trying to make sure that you get the right people. And we started off with a super talk from Katharina Schroeder on counter-enhanced catalysis, transitional catalysis, merging the two, photo-organocatalysis, lots of themes coming through in terms of organocatalysis and merging lots of new areas. Fabian then gave us a really excellent talk on an area I know very little about, synthetic plant glycans, complex synthesis, as I said at the time, of complex oligosaccharides, but really trying to unlock the mystery of plant glycans, you know, using both automated synthesis and normal synthesis, if you want to call it that. Rolf, then we had a super talk on the use of transitional metals, in particular palladium, in chemical biology, and how, you know, the use of palladium for the Suji-Trost reaction has now been very much honed for selective S carbon bond formation, where you could have 2 isomers, you're forming primarily one, and that took a lot of work to find the right ligand. But trying to get that working in vivo is still a work in progress, you know, trying to make sure you don't denature proteins, but, you know, really exciting results on that front. Melanie then talked to us a lot about repurposing enzymes. In fact, some of them were repurposed and they were even old enzymes, old yellow enzymes. So they must be very old at this stage if they've even got the description of being old. And really excellent work in terms of stereocomplementarity, being able to find 2 examples of those types of ligands that gives you exactly the opposite in enantiomeric products, and being able to merge them together by using chemical biology techniques. So that was really super. Again, more organocatalysis from Mario in terms of the isochalcogen ureas, collaborations with Andy Smith in St. Andrews, but done some excellent recent work all employing those alenoates. And certainly there's a lot of mileage and things to discover about that particular chemistry as well, in terms of how flexible it is and which products you get, and when is selenium better than sulfur and when is it the other way around. And then Fabian talked to us a lot about very fundamental inorganic chemistry, main group chemistry of phosphorus containing compounds, and making them pH and redox active ligands, and really been able to activate difficult to activate molecules, as we all know, like CO2 and SF6, easy for me to say. We finished up then with David, who talked to us about the preparation of functional organic molecules and wow, do they have function? They have lots of color and lots of clever chemistry going on there in terms of photo redox reactions, later photo writing, and even data storage, as we just discussed. So that's highlighting organic chemistry in Austria. I wish to thank my colleague on the EuChemS division, Michael Schnurk. He's been an excellent colleague for many years. He's been treasurer for the last number of years that I've been involved. And, you know, he really does put a lot of time and effort into the division. And I know he will be leaving that division soon, and look forward to his replacement and working with him. But ultimately, I'd like to thank the Austrian Chemical Society for helping with the organization of today and really putting the program together. And as I think, Melanie, you had said, you know, there's not often you get an opportunity where you meet other colleagues from Austria. So, you know, we all go abroad and we only meet each other a bit by bit. So having this type of focus is a very useful thing to have. And the fact that it's recorded it'll go up on YouTube and it'll be a snapshot really highlighting organic chemistry in Austria. So again, thank you for giving your time to this, and I hope you've felt it's of benefit, and I wish you all a good weekend. So thank you very much. Okay, so I shall stop recording.